What's up, Ninja Nerds? In this video today, we're going to be talking about diuretics. That includes every diuretic imaginable. We're going to talk about them. If you guys do like this video, you benefit from it, it makes sense to you, please help us. And the best way you can do that is by hitting that like button, commenting down in the comment section, and please subscribe. Also, I really highly suggest you guys check out our website. We'll have a link in the description box below. It'll take you there. We got some awesome notes, some amazing illustrations that our Ninja Nerd team has kind of put together. And I think it's really going to help you guys to enhance your learning experience. Get those illustrations, have the notes beside you, follow along with me as we go through and kind of write things down on the whiteboard. I really think it'll help. But without further ado, let's talk about diuretics. When we talk about diuretics, diuretics are basically, you're trying to be able to cause elimination of something. You're trying to eliminate sodium, you're trying to eliminate chloride, water, things like that in the urine. And the way that we have to do that is by targeting particular parts of the nephron. So we got to briefly, my friends, kind of go back over the anatomy, the physiology of some of the tubular reabsorption processes that are occurring. We're not going to go crazy. If you guys want to know way more about this, go watch that on our Renal Physiology playlist. But for right now, we have the different parts of the nephron. I just want to point out a couple parts here. One of these is this part here. So we kind of have what's called our Bowman's capsule. Then we go into this part here called the proximal convoluted tubule. The proximal convoluted tubule is extremely important. I'm going to denote this as the PCT. The proximal convoluted tubule is where a decent amount of sodium reabsorption occurs, right? So if you think about the actual sodium reabsorption that occurs within the proximal convoluted tubule, we'll represent sodium here as kind of a blue line. And then we'll represent what usually follows sodium because it generates an osmotic gradient. We'll represent that as water here in this kind of blue line here. So generally, what happens here is in the proximal convoluted tubule, you generally reabsorb a decent amount of sodium, and then subsequently you should reabsorb a decent amount of water in this section of the actual nephron. Approximately, if we really were to kind of approximate how much sodium is actually reabsorbed in the proximal convoluted tubule, it's somewhere around 65% of the sodium that gets reabsorbed here. So if you could imagine we did have a diuretic that directly inhibited sodium reabsorption, it would cause a massive loss of sodium in the urine and subsequently a massive loss of water in the urine. We don't actually have a very specific diuretic that targets directly sodium reabsorption and water reabsorption. It does it indirectly, but there is a diuretic that does work in the proximal convoluted tubule, and we'll talk about that one. But if we had a way of being able to inhibit sodium and water reabsorption directly, it would cause a massive loss of sodium into the urine and would cause a massive loss of water into the urine. Unfortunately, we don't have a drug that actually directly does that, but there is a drug that works to play some role in inhibiting some of the reabsorption processes that do occur in the proximal convoluted tubule. And we'll talk about that. It's called carbonic anhydrase inhibitors. So again, the first site of diuretic action here that is involved in diuresis is going to be at the proximal convoluted tubular level. And one of the drugs that specifically works there is going to be, let's actually do this one in red. This is going to be what's called your carbonic anhydrase inhibitors. Okay, so that's an important one. It doesn't directly block the reabsorption of sodium and water. It does something else. It does particularly something with bicarbonate, and it causes some sodium excretion and water excretion, but not a ton. Either way, that's the first site of the nephron. Now, generally, a lot of nutrients are also absorbed there, so glucose, amino acids, bicarbonate should be reabsorbed there, and that's a really, really important one. I'm going to represent that in black as well. So bicarbonate is a really, really important one that also gets reabsorbed in this section here, and that's really the thing that we're going to inhibit with the carbonic anhydrase inhibitors. But nonetheless, we go into the next part here. So then you go into what's called the descending limb of the loop of Henle. And if you guys remember, that plays a primary role in water reabsorption. So primarily, the only thing that really is kind of occurring here is water movement out of the descending limb of the loop of Henle. We're not going to really use any specific drug here. We'll talk about a drug that actually can kind of involve part of the proximal convoluted tubule and the descending limb. That's called mannitol or urea, but we'll talk about that one a little bit later, okay? It's kind of an interesting one. All right. Then we go from the loop of Henle, the descending, we go up. So now we're going into the ascending limb of the loop of Henle. When we get into the ascending limb of the loop of Henle, there is an actual very important drug that works here. And again, there is going to be some sodium reabsorption that occurs here, and there's going to be a decent amount of water reabsorption that occurs here in the ascending limb of the loop of Henle. So when we talked about this before, what we're really using for the proximal convoluted tubule, we'll talk about this a little bit later, is carbonic anhydrase that's inhibiting bicarb reabsorption. When we get into this part here in the ascending limb of the loop of Henle, we actually are directly blocking sodium and water reabsorption here. And again, what site is this again? This is the ascending limb of the loop of Henle. So this part here is called the ascending limb of the loop of Henle. Now, when we talk about sodium reabsorption in this section of the actual nephron, it's approximately 25%. So 25% of the sodium is reabsorbed in this section of the loop of Henle. And we do have a drug that directly blocks sodium and water reabsorption there. Very powerful diuretic. But again, there is a particular site there. What is that site again? That is over here. Another site of the diuretic action is called the ascending limb of the loop of Henle. And the particular drug that we will talk about that acts right there is going to be a drug called loop diuretics. So these are going to be called loop diuretics. That's their name, kind of working within the loop, right? So this will be your loop diuretics. All right, and we'll go over their mechanism of action in a little bit more detail. We're just basically kind of laying a foundation here. All right, then, as things continue to travel, so generally you'll have some type of filtration that occurs here at the glomerulus into the Bowman's capsule. So sodium and water will be moving through the proximal convoluted tubule. 65% of the sodium is reabsorbed here as well as water. If it's not reabsorbed here, it'll continue to move down the descending limb. Water reabsorption will occur in the descending limb. It'll go up the ascending limb. About 25% of the sodium and water should be absorbed in this section of the loop of Henle. Then if it doesn't get reabsorbed here, it continues into the distal convoluted tubule. So now we're going to move into the distal convoluted tubule. When we get to the distal convoluted tubule, there's actually 2 points here. So we have what's called the late distal convoluted tubule, and then we have over here the early distal convoluted tubule. In both of these sections, you are going to have some degree of sodium and water reabsorption. So there is going to be some sodium reabsorption that occurs here in the late and early distal convoluted tubule. And subsequently, there is also some water reabsorption that occurs in the early and the late distal convoluted tubule. We can actually inhibit the reabsorption directly in the early distal convoluted tubule and the late distal convoluted tubule. And we have drugs that specifically work there to block sodium and water reabsorption and block sodium and water reabsorption. What are those? Okay, so when we talk about the early distal convoluted tubule, this one actually accounts for about 5% of the sodium reabsorption. And then for the late distal convoluted tubule, it also accounts for about 5% of the sodium reabsorption. So that could kind of get you close to about 100% if you think about that, right? So 65 plus 25, so that would give you around potentially close to about 90%, and then you get an additional 5 and 10, so that's going to get around 100%. So generally that's where the sodium reabsorption and subsequently the water reabsorption are going to occur, based upon percentages. So when you think about it, the early distal convoluted tubule and late distal convoluted tubule, if they block sodium and water there, it'll cause sodium and water to be lost into the urine, but not as much as compared to the actual ascending limb of the loop of Henle. But nonetheless, what's the other 2 sites here that I want you guys to remember? Another site is called the early distal convoluted tubule. And again, the drug that works particularly at this site here is going to be what's called your thiazide diuretics. So your thiazide diuretics. And then the last one here is going to be at the late distal convoluted tubule. So this is going to be the late distal convoluted tubule. And this one is going to be your potassium-sparing diuretics. So this is going to be your potassium-sparing diuretics. And again, we'll go over these. There's 2 groups within this one. One are called aldosterone blockers and the other one are called ENaC blockers, but we'll go over them. But if you guys get the whole point here, if someone were to ask you just the basic point, okay, if we talk about the nephron, we know that there's different points along the nephron, such as the PCT, the loop of Henle, and the distal convoluted tubule, where sodium and water reabsorption occur. Now, at these particular sites, the percentage is different. Lots, 65% in the proximal convoluted tubule, 25% in the loop of Henle, and then 5% in the early distal convoluted tubule, and 5% in the late distal convoluted tubule. One of the big things to remember, I just want to add on here, is in the later part of the distal convoluted tubule, that sodium reabsorption is dependent upon the presence of antidiuretic hormones. So generally, in the distal convoluted tubule, especially in the late portions here, is very dependent upon aldosterone. So we're going to put here aldosterone. Okay, so very, very important here, is dependent upon the presence of aldosterone for sodium reabsorption and ADH for water reabsorption. But Nonetheless, we have an idea of the different parts where sodium and water reabsorption are occurring, and we know some of the sites by which these drugs actually work. One more that I did not mention is the osmotic diuretics. That's mannitol. So let's come over here to this small little diagram. So in this small diagram, what actually happens is mannitol is this very large kind of solute molecule. And what happens is whenever mannitol, it moves through the afferent arteriole, then it moves through the glomerulus, it actually gets filtered out across the glomerulus. When mannitol gets filtered out across the glomerulus, it's a decently sized kind of like large solute molecule. And what it'll do is it creates a very powerful osmotic gradient. And that powerful osmotic gradient will actually pull some water into the proximal convoluted tubule, and then it'll also continue to move down, so it's so large it can't be reabsorbed. It'll actually move down into the descending limb. And then it'll also cause a large reabsorption or large kind of like pulling of water into the tubular lumen here. And so what ends up happening is mannitol causes a massive diuresis, a massive degree of diuresis of primarily large amounts of water molecules. So that's one of the big things about mannitol, is mannitol does work at two particular sites in the kidney tubules. It's not primarily used as a diuretic. We'll talk about its actual indications a little bit later, because what it actually does is it's more particularly used for reducing intracranial pressure and reducing kind of acute increases in intraocular pressure. So we'll talk about that. But mannitol can work particularly at the proximal convoluted tubule, and it can work at the descending limb of the loop of Henle. So the drug that would actually work particularly at these two sites here to really cause a massive kind of like sucking or pulling of water into these actual tubules and then out into the urine is going to be your osmotic diuretics. Okay, and that's going to be things like mannitol and urea, and we'll talk about that a little bit more later. But we got the basic gist, right? So we have a basic gist of the different sections of the actual nephron, okay, where sodium water reabsorption is occurring, and we know where diuretics will specifically act at those sections. So we know proximal convoluted tubule is carbonic anhydrase, ascending limb of the Henle is loop diuretics, early DCT, thiazides, late DCT, potassium-sparing, and then technically osmotic diuretics can pull lots and lots of water into the proximal convoluted tubule and lots of water into the descending limb of the loop of Henle, and that can cause a massive loss of aquauresis, so it's going to cause a massive loss of water within the urine. And so that's one of the big things with osmotic diuretics. But now, let's get into a little bit more detail about the mechanism of action. So what we're going to do is we're going to zoom in on the proximal convoluted tubule, we're going to zoom in on the loop of Henle, the early DCT, the late DCT, and we're going to show you how these drugs actually help to promote some degree of excretion of sodium, chloride, water, other electrolytes, etc. Let's get into that. So first one is the proximal convoluted tubule. Now, when we actually talk about this, we're going to have some degree of filtration. So this is going to be from the Bowman's capsule, filtrates moving from the Bowman's capsule into the PCT. When things are moving here, the primary ion that we actually do care about that actually does get excreted across the glomerulus is something called bicarbonate. So we're going to have a molecule here called bicarbonate. Now, bicarbonate is a very interesting molecule. What happens is, in this proximal convoluted tubular cell, we also have another kind of like transport protein. And this transporter, basically what it does is, it's a sodium proton kind of antiporter. And what it'll do is it'll allow for sodium to move into the cell, all right, through this transporter. So sodium will come into the cell. When sodium comes into the cell, what also is going to happen is a proton is going to be pumped out of the cell. Okay, into the tubular lumen. So this is the tubular cell that we're zooming in on. This is the actual capillary blood where things can be absorbed, and this is going to be the lumen of the actual tubular cells. Okay, so this is the lumen of the tubular tissue, and this is actually going to be the tubular cell. This proximal convoluted tubular cell will excrete protons and reabsorb sodium, because again, these things are being filtered from the glomerulus. When they get filtered across the glomerulus, the sodium will actually get reabsorbed, protons will be excreted, and then bicarb will continue down here. When bicarb moves down, what it'll do is it'll combine with this proton here. So then what I'm going to do is I'm going to take this proton that I actually have that's now out here, and I'm going to combine it with my bicarb. When you take protons and bicarb, what do you actually get? The combination of these make something called carbonic acid, okay, called carbonic acid. Now, carbonic acid does dissociate, and when it dissociates into two particular molecules. One of these molecules is called CO2, and the other one is called water. Now, what happens is, in order for this process to occur, we have a very special enzyme that catalyzes and speeds up this particular step here. This dissociation of carbonic acid into CO2 and water is very highly catalyzed by this enzyme on the cell membrane. You know what this enzyme is called? Carbonic anhydrase. This enzyme here is called carbonic anhydrase. It's going to stimulate this step. Now, once it does that, when carbonic acid dissociates into CO2 and water, CO2 is lipid soluble. So bicarb is charged. It can't be reabsorbed. It's too charged. There's no specific transporter that allows for bicarb to be reabsorbed across this tubular cell. But if I turn bicarb indirectly into CO2, now I have a molecule that actually can be directly absorbed and dissolved and move passively across the cell membrane. So that's what CO2 is going to do. It's going to move across the cell membrane. Now, you know that there's water inside of cells. So what's going to happen is the water is going to combine with the CO2 inside of the cell. When it does that and these two actually combine, guess what they make? They make the same thing that happened here called carbonic acid. Guess who stimulates and catalyzes this particular step? There's a component of this enzyme in the cell. It's going to be carbonic anhydrase. Now, once I form that, now this thing is very unstable and it can easily dissociate. And guess what it can dissociate to, my friends? Protons and into bicarb. Now, this bicarb is essentially this bicarb that I wanted to reabsorb, but I couldn't do it directly. But now I have the bicarb in the cell, and there is a transporter on the basolateral membrane that can allow for me to get bicarb into the bloodstream. And when I push bicarb into the bloodstream, I move a negative ion out of the cell. So I got to bring a negative ion into the cell. So I bring chloride in, right? And then the same way, in order for sodium to be able to move into the cell and protons to move out of the cell, sodium has to move down its concentration gradient. In other words, I need sodium concentration in the cell to be low. And I need the sodium concentration outside the cell to be high. How do I do that? Well, good thing I got this pump back here on the basolateral membrane that pumps sodium out of the cell and pumps potassium into the cell. That's my sodium-potassium ATPase. So the sodium-potassium ATPase is what allows for my sodium to be on the lower end, which allows for sodium to be reabsorbed and then protons to be excreted via secondary active transport. And then the protons combine with the bicarb, make carbonic acid, dissociate into CO2 and water, CO2 gets absorbed, goes back and makes bicarb so that it can be reabsorbed. You're probably like, Zach, my head is hurting. How in the heck is this going to help me with diuresis? I got you. This is where the drug comes in. So now we're going to use the drugs called carbonic anhydrase inhibitors. And let's see now, after introducing this, so now that we understand the basic physiology, what we're going to do is these drugs are going to inhibit, so I'm going to draw this later here, I'm going to inhibit that particular enzyme. The carbonic anhydrase will now be inhibited. Okay, things get filtered. Sodium gets filtered, bicarb gets kind of filtered across. Generally what should happen is the bicarb should combine with the protons and make carbonic acid. Then carbonic acid should dissociate into CO2 and water. And then CO2 should be reabsorbed and then reformed into carbonic acid to actually help this process. Well, now what I'm going to do is I'm going to inhibit this enzyme. So now I'm no longer going to be able to stimulate the CO2 and water to make carbonic acid. So this process is now inhibited. That means I'm going to have less protons that I'm going to be able to excrete, less bicarb that I can reabsorb, means that less bicarb gets into the blood. If I don't pump protons out here across with this sodium transporter, am I going to be able to allow for sodium to come in if I'm not pumping protons out? No. So does sodium actually get reabsorbed here? No. So sodium won't get reabsorbed. So this blocks the sodium. So then sodium stays high out here as well. And I'm not going to be able to get the sodium into the cell. I'm not going to be able to pump the protons out. If I don't pump the protons out, can I combine a proton with bicarb to make carbonic acid? No. Then, if I don't have carbonic acid, can it dissociate into CO2 and water and then get reabsorbed? No. And so then I don't reabsorb CO2. I continually perpetuate this process. So the whole thing that happens here is I will not take bicarb and reabsorb it. Because if I inhibit this enzyme, both these, the actual one that catalyzes this process and this process, I won't be able to excrete the protons. And then if I can't excrete the protons, I can't convert this bicarb into carbonic acid and then into CO2 to reabsorb. So then I will not bring CO2 across the cell. I won't make carbonic acid. I won't reabsorb bicarb and I won't excrete protons. And then I won't allow for this reabsorption process to occur. So you see how it's kind of a perpetual cycle. So the end thing here is that because I can't actually form this thing here, this bicarb, if I don't make it this way, the bicarb stays in this form and then gets excreted out into the urine. So what's going to happen is I'm going to lose a lot of bicarb into my urine. A lot of bicarb. And very little bicarb will actually get reabsorbed. The other concept is that if I can't pump the protons out, I can't reabsorb the sodium inwards. So sodium will not get reabsorbed here. And so what will happen? The sodium will also be lost into the urine. And if sodium is lost into the urine, what should it pull with it? Water. Water should be pulled with the sodium as well. So when you think about the effect of a carbonic anhydrase inhibitor, it's going to cause diuresis, but the way that it's going to cause diuresis, it'll cause a little bit of sodium loss and a little bit of water loss. Nothing intense, but it's going to cause a massive bicarb loss. Let's put 3 arrows here just to kind of accentuate how insignificant that is. It's very significant, right? So carbonic anhydrases are going to inhibit this enzyme. The basic effect here is if you inhibit this enzyme, you inhibit the intracellular and the tubular kind of effect pathway. So if I inhibit this enzyme, I will not be able to take CO2 and water, make carbonic acid, make bicarb, make protons. If I don't reabsorb bicarb, my bicarb in the blood decreases. If I don't have protons, I can't excrete protons to combine with bicarb to make carbonic acid. So I don't make carbonic acid, I don't make CO2 and water, and I don't have CO2 to be reabsorbed. So bicarb then stays like this and gets excreted. On top of that, I also, even if some of this actual carbonic acid does form here, I can't actually convert it into CO2. And because I can't convert it into CO2 because I inhibited this enzyme here is inhibited via this carbonic anhydrase inhibitor, even if I do have some of this carbonic acid being formed because a little bit of the protons and a little bit of the bicarb do form, I'm not going to be able to allow for this to be converted into CO2 and water because I'm inhibiting this process. So either way, the whole concept is I'm not going to be able to reabsorb bicarb and I lose it into the urine. So it causes a massive loss of bicarbonate, a mild loss of sodium and water into the urine. That is the effect of carbonic anhydrase inhibitors. Okay? I hope that makes sense. Let's move on to the next one. So now we move on to the next one, which is the ascending limb of the loop of Henle. So this is talking about the loop diuretics. Now, how does the physiology occur here? Well, here we're going to have a tubular cell. So this is ascending limb, a loop of Henle cell. This is going to be the blood. So this is the peritubular capillary blood or the vasorectal blood. And this is going to be the lumen of the loop of Henle. Now, what happens in this site here is there's a special transporter here. You see this transporter here in blue? This is called the sodium-potassium 2-chloride cotransporter. Jeez. So what this does is this will push sodium into the cell. It'll push potassium into the cell, and it'll push chloride into the cell. Now, once the sodium is moved into the cell here, where will it go? Well, we want the sodium inside of the cell to be at lower concentrations so that sodium can easily move into the cell. So we want to allow for sodium to move down its concentration gradient. So the way that I'm going to do that is I'm going to take this sodium and I'm going to pump it out. And I'm going to use this thing called the sodium-potassium ATPase. And so this sodium-potassium ATPase will allow for sodium to be pumped out and potassium to be pumped in. Okay, that's pretty cool, right? The other concept here, so now I'm going to have lots and lots of potassium in the cell. So the potassium should build up because I got potassium coming in here, coming into the cell from the sodium-potassium 2-chloride cotransporter and potassium coming in across the sodium-potassium ATPases. All right, what about the chloride? The chloride, my friends, is going to come into the cell, but the chloride also has to be reabsorbed, and so the chloride will actually be reabsorbed across these channels, which may have some degree of like bicarb moving across. But either way, you're moving chloride into the blood, you're moving sodium into the blood, and generally what should naturally follow the sodium and chloride? Water. So you should allow for water reabsorption to occur here, okay? Now, what about this potassium? What's the potassium doing? Potassium's kind of interesting here. So there's lots and lots of potassium kind of sitting within the cell now, right? Because we had a lot of potassium coming in via these two pathways, the sodium-potassium 2-chloride cotransporter and the sodium-potassium ATPase, which is going to increase the potassium in the cell. So now potassium is going to be really, really high inside of the cell, and it's going to be lower in the tubular lumen. So what happens is some of the potassium will actually leak out via this kind of like little potassium transporter. And it'll leak some of the potassium out of the cell, causing the inside of the tubular lumen to become positively charged, right? Now, because of that positive charge here, there is other ions that are nearby. You know what these other ions are? You have other ions that are nearby, and these are called calcium and magnesium. Now, calcium and magnesium are positively charged. They don't want to be in the same vicinity as a lot of positively charged ions due to the potassium leaking out of the cell. So they don't want to stay in the tubular lumen. They're like, hey man, too much positivity here, I got to get out of here. And so what they do is they both move via the paracellular route in between the tubular cells and into the blood. So generally you should try to move calcium and try to move magnesium into the blood via this paracellular route, because what happens is this positive charge here is trying to inhibit them from moving forward throughout the tubular lumen. Okay? So that's the natural physiology that's occurring here in the ascending limb of the loop of Henle. Now insert loop diuretic. Okay. Loop diuretics come in, and what they do is the loop diuretics are going to specifically inhibit this transporter. So we're going to put in now the loop diuretics. How are they actually going to be helpful in this process? So loop diuretics are going to inhibit the sodium-potassium. So what are they going to inhibit? The sodium-potassium 2-chloride cotransporter. If I inhibit this transporter, can I bring sodium into the cell? No. So I prevent sodium from coming into the cell. Do I get sodium into the blood? No. So there's less sodium that gets reabsorbed here. Do I bring chloride across? No. I inhibit the chloride reabsorption. Do I bring chloride into the blood? No. So less chloride. If less sodium and less chloride are being reabsorbed, am I going to create an osmotic drag to pull water along with it? No, because I lost my sodium and chloride drag. So now there's going to be less water that's actually going to be pulled in this direction. So now what's going to happen is the sodium and the chloride are going to stay in the tubular lumen. So now I'm going to have lots of sodium that stays here, lots of chloride that stays here, and guess who's going to kind of follow along with them throughout the rest of the tubular tract? Lots of associated water. And so what I'm going to lose, if this were to continue through the tubular cells, let's say that it continued throughout the tubular cells, what am I going to lose into the actual urine then effectively? I'm going to lose in the urine lots of sodium. I'm going to lose lots of chloride. And I'm going to lose lots of water into the urine. Okay? Now, we're not done. This is where it's pretty cool. If I inhibit the sodium reabsorption, I don't have sodium available to move across the sodium-potassium ATPase. So if less sodium is moving across here, less potassium is moving across this pump. That means I'm going to have less potassium via this pathway. If I inhibit this pump, so I inhibited the chloride reabsorption, sodium reabsorption, what if I inhibit potassium reabsorption via this transporter? I don't bring as much potassium into the cell. And now there's less potassium via this pathway. So now the amount of potassium that's present in the cell is much less than normal. If there's much less potassium, that means there's not as much of a gradient for it to want to move out of the cell. That means that the positive charge is decreasing. There's less positive charge. So if you want to think about it, if there's less positive charge, there is more of a negative or relative negative charge. Do you think this negative charge is going to want to repel calcium and magnesium now? No. I'm not going to repel the calcium and magnesium as much now. And so since I'm not going to repel the calcium and magnesium from continuing to get reabsorbed, I'm going to not allow this reabsorption process to occur. So I'm not going to allow for calcium and magnesium to move across the tubular lumen, because now magnesium and calcium are attracted to the positive charge. So they want to stay in the tubular lumen. So they're going to continue throughout the tubular lumen and then get peed out. So what else will I see in the actual urine? I'll see lots of calcium and I'll see lots of magnesium. Oh my gosh, doesn't that make sense? So with that being said, when you think about loop diuretics, what you're actually getting here is the inhibition of this pump. But when you inhibit this particular co-transporter, subsequently, you're going to lead to less sodium, less chloride reabsorption, less water reabsorption. So now you're gonna have more sodium, more chloride and water in the urine. If I inhibit these, I inhibit the movement of potassium into the cell. So I have less potassium to leave the cell, less kind of repulsion of the magnesium and calcium. So now it's not gonna be reabsorbed. It's gonna stay in the tubular lumen where the negative charge is, and you're gonna pee that out too. And you're gonna lose lots of calcium and magnesium. So that's a really cool concept about loop diuretics and how it's going to help you to kind of cause this diuretic effect. All right, so far we've covered carbonic anhydrase inhibitors, we've covered the loop diuretics, we've covered the two sections they work at. Let's now move into the distal convoluted tubule. All right, so the next one, early distal convoluted tubule. What's gonna happen here? We gotta do basic physiology. I know it's annoying, but it's really helpful because once you understand the phys, you just basically plug in the drug and then just oppose everything else. It's very simple, right? So as we have things coming through, so we were moving through what part? So we were moving through kind of in a systematic fashion here so that you guys understand here. We're at this point where we filtered things out of the actual, Bowman's capsule. We went through the proximal convoluted tubule. We went through the descending limb. We went through the ascending limb, and now we're at this portion right here. And then we'll go into the collecting duct, right? So far we talked about carbonic anhydrase inhibitors, inhibiting bicarb, sodium reabsorption, and leading to a little bit of water loss. We talked about how loop diuretics will inhibit sodium, potassium, chloride co-transporters, and how it'll lead to sodium loss, chloride loss, water loss. On top of that, you also inhibit calcium and magnesium reabsorption. Now we're going here to how the sodium and chloride reabsorption is being inhibited at this spot, and then also water reabsorption. So as things are coming through here, so you should have some movement of particular ions as we're kind of coming from this point here to here. Okay, so we're going to have some ions or things moving through this area. So particularly, it's going to be sodium, chloride, water, and some other things. So what happens is you get to this point here, and there's a special transporter here. It's called a sodium chloride co-transporter. And what it's going to do is it's going to take sodium that's here, and it's going to move it across this transporter. Subsequently, you're also going to have some chloride. And the chloride should move across this particular transporter. And then sodium will then enter into the cell, usually kind of down its concentration gradient. So now sodium and chloride should be in this cell. It's actually just for the sake of kind of like putting this here, just because I have the diagrams here. We're going to put chloride up here, sodium here, but either way, it's the same concept. We get sodium and we get chloride into the cell. Well, then what happens is the chloride has to get out of the cell and into the bloodstream via this chloride kind of transporter, which will put chloride into the bloodstream here. Then sodium has to move out of the cell and into the bloodstream, and it does that via this sodium potassium ATPase, right? So that's how we get the sodium into the bloodstream. Now, subsequently, if you're pulling sodium and chloride across this tubule, you're also going to subsequently be dragging what with it? Water. So you should also drag some degree of water into the actual blood. Because again, what we're doing here is this is going to be the early distal convoluted tubular cell. This is the blood, and this is the lumen of the distal convoluted tubule, early part. We're pulling sodium across here, we're pulling chloride across here, and we're pulling water across here, right? So effectively, that's what you should be doing, is increasing the chloride reabsorption, sodium reabsorption, and water reabsorption. Another thing that I wanted to quickly add on here, because thiazides play a role within this as well, and it's very important because we'll talk about it when it comes to like indications and adverse effects, but there's another particular ion that gets filtered. So we talked about sodium, we talked about chloride, we talked about water, but there's also an important ion here called calcium. Now, calcium reabsorption in the distal convoluted tubule is dependent upon a particular hormone. We're not going to go over this kind of like in crazy detail, but there is a particular hormone here that does regulate the activity of the calcium channel here, and this is called parathyroid hormone. So it does help to kind of like control the absorption of calcium. But nonetheless, let's say that the calcium absorption is occurring with the presence of PTH. When calcium gets absorbed and it moves across the actual tubular lumen into the tubular cell, the question is okay, how does the calcium get out? So there's a transporter on the basolateral membrane. It's a really cool one. And what it does, it pumps the calcium into the blood. And then what it does is generally, what you should be doing is if you're bringing sodium into the cell, and then you're using the sodium potassium ATPs to pump the sodium out of the cell, the sodium concentration in the cell theoretically should be lower. So then what happens is sodium will move down its concentration gradient into the cell, which allows for calcium to move out of the cell. So there's a sodium-calcium exchange, if you will. Now, here's where thiazides, now just throw a wrench in this whole physiology process by saying, okay, I'm gonna give the patient a thiazide diuretic. How does this actually help? How does it work, my friends? So thiazide diuretics are going to inhibit this sodium chloride co-transporter. If I inhibit the sodium chloride co-transporter, do I bring sodium and chloride into the cell? No. So I inhibit sodium and chloride reabsorption. If I inhibit sodium and chloride reabsorption, I have less chloride in the cell to move out into the blood. So I decrease my chloride that's actually getting reabsorbed. If I have less sodium being brought into the cell, I have less sodium to pump out via the sodium potassium ATPases, and I have less sodium that moves into the blood. If I have less chloride and less sodium being reabsorbed across the tubular cell, I don't have as much water to reabsorb across that tubular cell by that osmotic gradient that sodium and chloride creates. So therefore, less water is reabsorbed across the tubular cell. So now think about that. If that's the case, then what am I losing into the patient's urine? I'm losing sodium, decent amount of sodium. I'm losing chloride, decent amount of chloride. And I'm losing water. Now, the other concept here that's really cool is how does this affect calcium? Because we already know that loop diuretics called calcium loss, right, and magnesium loss. Well, thiazide diuretics do something really interesting. Okay, if I inhibit the sodium chloride co-transporter, I inhibit sodium from coming into the cell, right? And now if there's less sodium that's coming into the cell, then what does that do to the amount of sodium concentration in the cell then? I have less sodium being brought into the cell. That effectively will lower my sodium concentration in the cell. You know what that's gonna do? Remember I told you that sodium moves down its concentration gradient through this exchanger. So now if sodium is moving from high concentration to even lower concentration here, what's going to happen? I'm going to bring more sodium into the cell. If I bring more sodium into the cell, I have to allow for more calcium to be exchanged out of the cell. So that means when I bring the calcium in, I'm going to I'm going to increase my absorption of calcium, and I'm going to cause a ton of calcium to be exchanged across the sodium-calcium exchanger. Theoretically, what does that do to the amount of calcium that goes into my bloodstream then? I increase the calcium absorption. Isn't that cool? So the whole concept I want you to understand here is when you inhibit the sodium chloride co-transporter, you inhibit sodium and chloride reabsorption. That causes it to get lost in the urine. But you also pull water with it. When you inhibit this transporter, you don't bring as much sodium into the cell. That makes the sodium concentration in the cell lower. Now the sodium that wants to move down its concentration gradient via this exchanger is going to move more powerfully and more consistently. And if you have more sodium coming in, you have to have an equal amount of calcium coming out. So if a lot of sodium comes in, a lot of calcium goes out. And that means the calcium concentration in the cell gets lower, and that pulls more calcium into the actual tubular lumen. So as you pull more calcium out, you're going to theoretically, as you pull more of this calcium out, lower the calcium level in the cell, which is going to cause an increase in the calcium reabsorption across this cell, an increased exchange via the sodium-calcium exchanger, and cause an increase in calcium in the blood. And so what will happen to the calcium here? You'll have less calcium present within the urine. It's pretty cool, right? All right. But that's how thiazide diuretics work. Let's move on to the next part here. So we move into the late distal convoluted tubule. Now, if you kind of remember our diagram here, at this point we've covered carbonic anhydrase inhibitors, we've covered loop diuretics, we've covered thiazide diuretics. So now we've covered 65% of the sodium reabsorption, 25%, 5%, and now we have the remaining portion here. If we get to the later distal convoluted tubule, we're going to inhibit the remaining 5% of sodium chloride reabsorption and subsequently water reabsorption at this site. So let's now zoom in on that segment and look at it. All right, basic physiology here. When you have things moving through here, so again, we're going to have some degree of sodium, some degree of chloride, some degree of water, and things like that moving down through this actual tubular lumen. Here's going to be the distal convoluted tubular cell, the late part, and then here's going to be the blood, where it should be around the peritubular capillary blood. All right. This section here, we have different types of cells. So you have principal cells, and then you have intercalated cells. Technically, this is what's called, if you really want to remember, this is technically what's called a principal cell. So principal cells are involved for electrolyte balance. Intercalated cells are involved in acid-base balance. We'll talk about that a little bit later. But what I want you to remember here is this principal cell is very dependent to some degree on aldosterone. So you guys remember that we have our adrenal cortex, and our adrenal cortex makes a particular molecule called aldosterone. Now when aldosterone is secreted, it's a steroid hormone. And what I'm going to do is I'm going to represent aldosterone by that circle there, okay? When aldosterone is secreted, it moves through the blood, and then it can cross directly across the cell membrane because it's a steroid type of hormone and bind with a particular receptor. That receptor that it binds to will then click in to the DNA and activate particular genes in the DNA. And then when you activate these particular genes, this will increase the synthesis of particular types of proteins. One of the proteins, let's actually represent this in red, one of the proteins that it actually synthesizes is called a sodium channel. So here's one of the proteins. The second channel it synthesizes is a sodium-potassium ATPase. And the third channel is a potassium channel on the apical cell membrane. Okay? So there's three channels that will help to synthesize in the presence of aldosterone. So again, aldosterone will bind onto an intracellular receptor, activate a transcription factor, stimulate the genes to make particular proteins. Now, once these proteins are activated and expressed, we now have a channel here that's open. Usually these are not there in very powerful amounts, but whenever you have aldosterone, it's going to increase the expression of this. Now, once this channel's here, this is called a ENaC channel, so an epithelial sodium channel. When sodium is present at this portion here, what it's going to do is it's going to move via this channel from the tubular lumen into the tubular cell. Once the sodium is brought into the tubular cell, we want to get it into the bloodstream to reabsorb the sodium because that's where 5% of the sodium reabsorption occur. So in order for me to pump the sodium via this channel here, I have to pump potassium inwards. So this is via the sodium-potassium ATPase, and this pumps the sodium into the blood. All right? But it pumps potassium from the actual blood into the cellular tissue. The next thing that happens here is I have this potassium in the cell that I'm actually bringing into the cell from the blood. That potassium can actually, that's right here, right? We're going to have this potassium. It's going to be right here now. It's in the cell. It's going to want to move down its concentration gradient, so it's going to want to move out of the cell into the tubular lumen. And so then I'm going to excrete some of the potassium out of the actual cell and into the urine, right? So that's the concept there. Now generally, when I reabsorb sodium across this cell, what would I also subsequently pull with me if I have this process? So if I reabsorb sodium, I should subsequently pull what other molecule into the particular bloodstream, as long as ADH is present? Water. So this is generally what should actually happen here. Now, what I'm going to do is, and I should actually have less potassium within the bloodstream, right? So the other thing is I should have less potassium. Let's put this one up here. I should have less potassium within the bloodstream because I'm excreting the potassium. I'm pumping it into the cell and then pumping it into the tubular lumen. So there should be less potassium in the actual blood. But I should reabsorb sodium, reabsorb water, and excrete potassium, usually in the presence of aldosterone. Now what I'm going to do is I can have two drugs. One of the drugs is what's called an aldosterone. So I have one drug category for the potassium-sparing diuretics. There's two drug categories. One is an aldosterone antagonist. So one is called an aldosterone antagonist. The second potassium-sparing diuretics are called ENaC blockers. So I have aldosterone blockers and ENaC blockers. All right, how does this work? If I give an aldosterone antagonist, what this will do is it'll inhibit this process here. It'll block the aldosterone from being able to bind onto the receptor and activate transcription factors. It'll then inhibit this particular process. So now you won't be able to bind onto the genes and activate the synthesis of particular genes to make particular proteins such as ENaC channels, potassium channels, and sodium-potassium pumps. So then I'm going to inhibit the potassium pump from being formed, sodium-potassium pump. I'm going to inhibit the ENaC channels from being formed, and I'm going to inhibit the potassium channels from being formed. If that happens, will I have sodium channels available to reabsorb sodium? No. So then I don't reabsorb sodium. If I don't reabsorb sodium and I don't have the sodium-potassium pumps available to pump the sodium out of the cell and into the blood, what's going to happen to the sodium inside of the blood? It's going to decrease. If I don't reabsorb sodium across the actual tubular lumen, tubular cell into the blood, am I going to subsequently pull water in the presence of ADH across? No. So then I don't reabsorb water. If my sodium-potassium pump is inhibited, not only do I not pull sodium into the blood, but I don't pull potassium into the cell. So then I have less potassium available into the cell. On top of that, I inhibit this particular channel. And so now I can't excrete potassium into the actual urine. And so I don't lose potassium into the urine. So what happens to the potassium within the blood? Well, I can't pump it into the cell and I can't pump it out of the cell. So the potassium within the blood increases. This is why it's called a potassium-sparing diuretic, because you're not going to reabsorb sodium, and so therefore you won't reabsorb water effectively. So what am I going to lose within the urine? I'm going to lose some sodium, and I'm going to lose some water. But what will I not lose? I will not lose potassium. So there will be very little potassium present within the urine. So that's a really cool concept. Well, that's how aldosterone antagonists work. What about the other category of potassium-sparing diuretics, such as ENaC blockers? Believe it or not, it's the same concept. If I, think about this, block the ENaC channels, okay? So I block this ENaC channel. Can I bring sodium into the cell? No. If I can't bring sodium into the cell, now I don't have sodium available in the cell. So I have less sodium to pump out of the cell, and subsequently less potassium available to pump into the cell. So the same concept here will occur, that I'll have less potassium coming into the cell, and I'll have less sodium moving across the sodium-potassium pump. Because again, all the ENaC blockers do is block this channel. You'll still have this channel and you'll still have this channel. But look what it does indirectly. You block this channel, you have less sodium. If I have less sodium, I have less sodium to move across this sodium-potassium pump. So less sodium gets reabsorbed. Plus, if I reabsorb less sodium, I don't push as much potassium into the cell. So I have less potassium entering into the cell. If I have less potassium entering into the cell, even though this channel is still open, there's going to be less potassium excreted. So there will be some degree of potassium loss, even with these ENaC blockers. So you'll still have potassium that'll be lost. But look how little potassium will actually be lost. Very little potassium will be lost because you have very little potassium that's got pumped into the cell, therefore less intracellular potassium. That means that there's less potassium to move down its concentration gradient out into the urine. So you see how ENaC blockers indirectly decrease potassium excretion and again, reduce sodium reabsorption, water reabsorption, and cause sodium and water wasting, but not potassium wasting. That is the cool concept with these drugs. So that is how the, again, potassium-sparing diuretics such as the aldosterone antagonists and ENaC blockers particularly work within the nephron. So at this point, my friends, we have covered carbonic anhydrase inhibitors, loop diuretics, thiazide diuretics, potassium-sparing diuretics, and we even briefly discussed the osmotic diuretics like mannitol and urea and really how they work in the proximal convoluted tubule and how they work in the descending limb loop of Henle, and they're very large impermeable molecules that suck water into the tubular cells, in the tubular lumen and cause massive aquauresis. Not really sodium loss, aquauresis, massive, massive loss of electrolyte-free water. And that can really cause significant dehydration. But again, that's how mannitol and urea would work, which are the osmotic diuretics. Now what we need to do is, now that we've built the mechanism of action, let's talk about how do these particular mechanisms of causing bicarb loss, sodium loss, water loss, all this type of loss, all this type of loss, all this type of loss, help people in particular disease states, one by one with each type of diuretic. And then what we'll do is, we'll talk about the main indication of diuretics, which is fluid diuresis. How do we actually specifically attack and approach fluid diuresis? And then later we'll go into the adverse effects of each drug category. Let's get into it. All right, my friends, so let's start off with the first category of diuretics that we talked about a little bit, which is the osmotic diuretics. This is mannitol and urea. All right, so those are the two particular drugs that I want you to remember for this category. And again, we talked about how their mechanism of action is not really designed particularly as a diuretic, so fluid diuresis. They really can cause massive loss of water in your urine, but that's not their true indication. But nonetheless, we talked about how they work in the proximal convoluted tubule and the descending limb of loop of Henle and really pull a lot of water into the kidney tubules. But you know what else they do? So let's say that we have mannitol urea and we get this molecule. It's a very large kind of molecule that you can actually kind of creates a lot of osmolarity. So what this really does is it really increases the osmolarity. So it really kind of increases the osmolarity of the blood. Now, when you increase the osmolarity of the blood or the osmolality of the blood, what you do is you make the blood kind of create this very strong gradient to pull water into it. So this would be something like mannitol or urea. They're very large solute molecules, especially mannitol. And what it's going to be primarily doing is creating an osmotic gradient that pulls water from extravascular spaces. So basically it's going to pull it from things like tissue cells or interstitial spaces. So from in the cells and the interstitial spaces, it's going to yank tons and tons of water. So what it'll do is it'll yank water from areas of tissue such as brain tissue or such as things like the vitreous humor in the eye. And it'll rip tons of water into the actual bloodstream. So effectively, when you think about this, what the mannitol is actually going to do, or the urea is going to do, specifically, I would actually want you to primarily remember mannitol is the most commonly utilized one. It's really going to increase the water in your vascular space and pull tons of water from tissue such as the brain tissue and such as the vitreous humor. Now, why would that be beneficial to pull tons of water and dehydrate your cells in your interstitial spaces and the vitreous humor in the eye and the brain tissue? Here's why. In patients who have a lot of cerebral edema, so they have what's called increasing intracranial pressure, what you want to do is reduce the pressure inside of the brain. So oftentimes this can be to big brain kind of like generally sometimes large strokes, so large like ischemic strokes or very large hemorrhagic strokes or subarachnoid hemorrhages or cerebral edema in situations of diabetic ketoacidosis. But either way, lots of cerebral edema. What you can do is you can reduce some of the edema in the brain tissue by pulling water from the interstitial spaces in the brain and from the actual brain tissue and shrink the actual brain to reduce the intracranial pressure. So that is one particular reason that we could utilize this drug, is to help to effectively pull water, which what will this do? Help to lower ICP. Okay, so that's particularly the indication. The other situation is when patients have very high pressure in their eye. Okay, so in situations of where maybe they have acute glaucoma or increased intraocular pressure, whenever they have very, very high intraocular pressures, what you can do is you can give mannitol and it'll help to be able to yank some of the fluid from the vitreous humor, which is in the posterior segment of the eye, and that'll help to kind of shrink that portion and decrease the pressure inside of the actual ocular socket. And so what this may do is this may decrease the intraocular pressure. So this could be used in situations such as cerebral edema. So the indication of this in the brain tissue is it may be particularly for cerebral edema, any type of cerebral edema, and we're going to try to reduce the ICP, or it could be utilized in situations such as glaucoma, acute glaucoma, where there's high intraocular pressure and you're trying to pull fluid from those tissues. Makes sense, right? Now, when you pull the fluid from these tissues, you significantly increase your actual plasma blood volume, because this mannitol creates a very powerful osmotic gradient that yanks a lot of water into the actual vasculature. So then imagine now you have a ton of water that's moving through your vascular space. Then what happens is, if this moves into the glomerulus and gets filtered easily across the glomerulus, what are you going to filter across the glomerulus? You're going to filter across this mannitol. So that's one thing. You're going to filter across the mannitol, across the glomerulus. But what else are you going to filter across? Tons and tons and tons of water. So what else will I have? Tons of water that I yanked from my tissue spaces. And so this is going to cause a massive volume of water that's getting filtered across the actual glomerulus. But on top of that, I'm also going to have mannitol in the kidney tubules. What did I tell you that mannitol does in the kidney tubule, specifically in the proximal convoluted tubule? So in two parts, the proximal convoluted tubule and the descending limb of the loop of Henle. Now, it's going to yank tons of water from the interstitial spaces and from the cells of the proximal convoluted tubule and the distal limb of the loop of Henle, and then what is it going to do? It's going to cause a massive loss of water. So it's going to cause massive aqua uresis. So what is this going to be containing? Tons and tons and tons of water will be lost. This will be water that came from the tissue spaces in the brain, tissue spaces of the vitreous humor, other tissues within the body, and water that got pulled from the proximal convoluted tubular cells and from the descending limb of the loop of Henle and the interstitial spaces around that. So it's going to cause massive water loss. This is called aqua uresis. Now, very briefly, I want to just quickly talk about some of the complications that you can have from this. Well, especially since you're losing like a massive amount of water from your body. You're really draining your intravascular volume. So if your kidneys are functioning and all of this blood volume that you kind of have now from pulling water from these tissue cells, and it's in the vascular space and your kidneys are completely functioning, it'll filter all that water off and more mannitol into the kidney tubules and really drain your blood volume. So effectively, what this could potentially lead to adverse effect-wise is this could cause significant volume drop. So this can cause hypovolemia, sometimes to the point where it can actually even significantly drop the patient's blood pressure. The other concept here is if you lose tons and tons and tons of water across your actual kidneys, what else could it do? You're dropping the water, so what this also could do is this could actually cause a loss of water in comparison to the actual sodium inside of your blood. And so what this could actually do is this can cause hypernatremia within the blood. So watch out for hypernatremia as another potential complication. Now, this is if the kidneys are intact. So what I want you to remember is this is particularly going to be happening if the kidneys are good. They're good to go. They're functioning well. So all the volume that you're actually pulling from the tissue spaces, you're peeing out. So it can lead to hypovolemia and hypernatremia due to the excessive loss of water, and then now your sodium is going to go up because you lost a ton of water. So it's kind of like a relative hypernatremia. If the kidney tubules are not intact or your kidneys are not functioning, here's where it's bad. So now your kidneys aren't functioning. They're all jacked up. If these puppies are all jacked up, now the water that you're supposed to be taking from the all this kind of vascular space and kind of excreting it out, this is all lost. So this water here that you're supposed to be taking and peeing out, you can't do it anymore. You're now losing that ability. So now all this volume stays in your vascular space. If the kidneys are if you have renal failure and you give the mannitol to them, it'll massively increase your actual blood volume. And you know what that'll do? That'll cause some of this fluid to leak out of the vasculature and into particular tissue spaces. And you want to know what one of the most common tissue spaces it'll leak out into? The lungs. And it'll cause massive pulmonary edema. So I want you to watch out for that if the patient has some type of acute renal failure, or if they have end-stage chronic kidney disease and they aren't able to excrete the water that they pulled from their tissue spaces into the blood volume, or they weren't able to allow for the mannitol to pull some of the water out via the kidney tubules and then cause them to release cause aquuresis, that fluid will stay in the vascular space, leak out into the pulmonary interstitium, and cause pulmonary edema. So these are the things that I want you to watch out for. Also, this is weird, right? It can cause hypernatremia and hypovolemia if your kidneys are functioning. If your kidneys are not functioning, it can cause the blood volume to massively increase. So it's almost opposite of this one. And then if your blood volume massively increases, it'll cause pulmonary edema. But if you hold on to all of that water and you can't pee it out, what happens to the relative relationship between sodium and water now? Now you're having a lot of water and a relative decrease in the amount of sodium, so it can cause hyponatremia. So watch out for that. I know it may seem slightly confusing, but watch out that whenever the kidneys are not working, it may cause hyponatremia within the blood and pulmonary edema if the kidneys are not functioning. If the kidneys are functioning, it'll cause hypovolemia and hypernatremia. Okay, that's mannitol and urea. Primarily mannitol given in situations of ICP crisis, intraocular pressure crises to reduce cerebral edema, acute glaucoma. If we give these medications, they're going to cause massive loss of water into the actual urine if the kidneys are functional, which can lead to decrease in blood volume, dehydration, and a relative hypernatremia. If your kidneys are not functioning, they can't excrete all of that large volume of fluid and water that's in your vascular space, they stay in the vasculature because you can't pee it out, and then it causes pulmonary edema and a relative hyponatremia. All right, now let's move on to the next drug category. All right, my friends, so carbonic anhydrase inhibitors. What are the particular drugs in this category? The main one that I want you to remember is acetazolamide. Really, that's the only one, I think, actually. So acetazolamide is the big one here. When we talk about carbonic anhydrase inhibitors, we know that they act at what particular section of the actual kidney tubules, or the nephron. They specifically work at the proximal convoluted tubule. But do you remember, 65% of sodium is reabsorbed in the proximal convoluted tubule, but it doesn't directly inhibit sodium reabsorption. It inhibits the carbonic anhydrase enzyme. And what that does is if you inhibit the carbonic anhydrase enzyme, you don't reabsorb what? Bicarb. And so what do you lose a lot into the actual urine? Bicarb. But you also, if you inhibit the carbonic anhydrase enzyme, you also do lead to less proton excretion and less sodium reabsorption. So you do lose a little bit of sodium in the urine, and subsequently you'll lose a little bit of water as well. But the main thing that you lose is a lot of bicarb. Now, if you lose bicarb in your urine, what happens to the amount of bicarbonate within the blood now? Because you're not reabsorbing the bicarb. So you're basically, this process where you're supposed to be pulling bicarb into the blood is decreased. So what happens to the bicarbonate level within the blood? It's going to be lower. If the bicarbonate level in the blood is lower due to not a lung issue, but due to an actual kidney tubule issue, then what is that called? What happens if your bicarb is low? This will do what to your pH? This will subsequently increase your, this will decrease your pH. This will decrease your pH. Because again, the reason why is if you have less bicarb, you have less base. And so you have more of the protons around to be able to not be bound up with the bicarb. Because you remember this equation, right? That we take basically protons plus the bicarb, and when you tie them up, it makes something called carbonic acid, and that disassociates into CO2 and water. Well, if we have less of this bicarb, we're going to have more of the protons over here present. And so that's one of the downsides here. So if we have less of the bicarb, we're going to have more of the protons that won't be complex with the bicarb. And what's the high protons going to do? It's going to drop the patient's pH. And so this leads to a condition called metabolic acidosis. But it's not due to an increase in the anion gap. So we call this a non-anion gap metabolic acidosis. Now, why is that actually kind of helpful? Is that helpful? There is a benefit to it. One of the benefits to this is that whenever you have this lower amounts of bicarb, and it actually causes the pH to become more acidic, what this does is this acts on particular chemoreceptors, what's called central chemoreceptors. And when you act on these central chemoreceptors, you actually stimulate these central chemoreceptors. And what they do is they increase the actual rate of respiration and the depth of respiration. So now, because it's acidic, because your blood is acidic, you want to be able to try to make the pH go up. And so what it'll do is it'll increase the actual flow to the actual lungs. And what you're going to try to do is you're going to try to increase the respiratory rate and the depth. So you're going to try to breathe faster and deeper. And the whole point to this is there's 2 points. You're going to increase your tidal volumes, right? Your minute ventilation is going to increase. And so hopefully you'll bring in more oxygen to come across and actually move across the lungs and into the actual blood. So hopefully with increasing your tidal volumes, you'll improve your oxygenation. So that's one theory behind that, is that you'll increase the amount of oxygen that gets absorbed across. But the other thing is that you'll blow off more CO2. So more oxygen will move across the blood, but also, so more O2 will be inhaled, but there'll also be more CO2 that's exhaled. And if you blow off more CO2, then what's going to happen to the CO2 level within the blood? Then the CO2 level within the blood is going to decrease. If there's less CO2, what happens is you're going to shift this reaction to the right and then decrease the amount of protons. And so that'll help to be able to kind of create what's called a respiratory alkalosis. So the effect here is that you're trying to make the patient breathe a little bit faster. And this creates something called a respiratory alkalosis. So you're trying to make the patient breathe at a faster rate to increase their oxygen inhalation, but also try to make their pH go up. Okay? So it's trying to compensate for the low pH that you see with acetazolamide. Why in the world would this be helpful? There's one particular indication, and this is in patients who have increased risk of altitude sickness. And altitude sickness, or basically they develop what's called high altitude pulmonary edema. So whether it's altitude sickness or another condition related to it called high altitude pulmonary edema, you can give this particular drug. Because when you give this drug, as they go up to higher altitudes, there's less availability of oxygen. And so they can develop kind of a hypoxemia, and sometimes they can develop into pulmonary edema. If you give them acetazolamide, prior to them actually kind of ascending, it can kind of get their blood a little bit more acidic, which increases their tidal volumes, their respiratory rate, their respiratory depth, to be able to blow off some of the CO2 to make their pH kind of a little bit increase to compensate, but also bring in more oxygen to improve their actual ventilation and also hopefully improve their oxygenation. So this is one of the things that we can actually utilize. It's actually pretty cool. There's one more indication for it, and we'll talk about it a little bit later. So one of the indications of causing this metabolic acidosis is treating altitude sickness. The other one is we can use this in what's called diuresis. So we can use this in a process called fluid diuresis. It's kind of an adjunct of therapy, and we'll talk about how we actually do it. What it does is it treats a patient who has high pH. So patients who have what's called a metabolic alkalosis, we can give this drug because it will decrease the pH. We'll talk about it a little bit later. All right, what else can acetazolamide do? So this is related to the diuretic effect, right? So this will actually cause help with diuresis and altitude sickness via the diuretic effect. There is another effect that this drug may have that has nothing to do with the diuresis. It's what it can do on kind of preventing or reducing the production of particular fluids. What it can do is it can work on the eye, and it can actually decrease the kind of what's called the aqueous humor production. So it can reduce the aqueous humor production. And if we reduce the aqueous humor production, we reduce the amount of fluid that's present within the kind of the anterior chamber of the eye, which can help to reduce intraocular pressure. So it can reduce intraocular pressure, especially in states like glaucoma. The other thing it can do is it can also work particularly within the ependymal cells, within the who make cerebrospinal fluid, and it can actually decrease CSF production. And if it decreases CSF production, that may be beneficial in patients who have high intracranial pressure. So high intracranial pressure, that is, but it's actually not due to a primary kind of or it's not due to any kind of disease, not due to a disease process. Like there's no kind of a structural lesion or obstruction or any kind of process like that. So it's a disease that's actually idiopathic. You've excluded every other cause that's causing their intracranial pressure to be high, and it's called idiopathic intracranial hypertension, also known as pseudotumor cerebri. So this is really what it's actually going to treat. It's going to reduce the CSF production in patients who have ICP that's not due to an actual disease process, it's idiopathic. And because of that, or pseudotumor cerebri, it will actually reduce the CSF production and reduce the intracranial pressure. So it'll actually reduce the ICP. All right. So when we talk about carbonic anhydrase inhibitors or acetazolamide, it's actually going to cause a lot of bicarbonate excretion into the urine, mild amount of sodium and water excretion, but because it kind of really causes a lot of bicarb excretion in the urine, it reduces the amount of bicarb in your blood. That causes a metabolic acidosis, not anion gap related. Because it causes an acidosis, that can actually increase your chemoreceptor sensitivity and cause your chemoreceptors to activate your respiratory center to breathe at a faster rate and depth. So you hyperventilate to breathe in more oxygen to improve your oxygenation and breathe out more CO2 to be able to kind of compensate for the acidosis and create a respiratory alkalosis to compensate. This can be used in altitude sickness, high altitude pulmonary edema, as well as a diuretic effect to treat what's called metabolic alkalosis with loop diuretics and thiazide diuretics. It also reduces aqueous humor production to reduce intraocular pressure and glaucoma, and reduces CSF production to produce ICP in patients who have no underlying lesion or disease process that causes their ICP to build up. It's called idiopathic intracranial hypertension. Okay, now that we talked about these ones, let's move on to the next category of drug called loop diuretics. All right, let's talk about diuretics, particularly loop diuretics at this point. So we talked about kind of your osmotic diuretics, we talked about the carbonic anhydrase inhibitors. Now let's hit particularly the loop diuretics. So names, we already know that with these ones, they're the, kind of cheated a little bit, furosemide. So you got your furosemide here. This is probably gonna be the most commonly utilized one, also known as Lasix. Then you have your torsemide. Bumetanide's also a pretty good one, very, very powerful one. And then the last one here is gonna be ethacrenic acid. And I repeat, ethacrenic acid. Now, with this particular drugs here, with the loop diuretics, what I want you guys to think about here is that they are very, very effective at causing a massive amount of sodium and water loss into your urine. We know that because we talked about how they work at the ascending limb of the loop of Henle, and they directly block the sodium potassium 2 chloride cotransport that causes a massive loss of sodium, massive loss of chloride, massive loss of water into the urine. So they're the most powerful diuretic. In theory, if there was a diuretic that directly blocked sodium and water reabsorption in the proximal convoluted tubule, that would be the most powerful diuretic, but we don't have one. And if you're thinking it's carbonic anhydrase inhibitors, remember I told you it does not directly block sodium and water reabsorption. It blocks bicarbonate reabsorption, which causes a little bit of sodium and water loss. But loop diuretics are very powerful, so they can cause a lot of fluid loss. So that's really good when you want to take fluid off of somebody who is in congestive heart failure, or they're in chronic kidney disease and acute kidney injury and they're not making much urine, or they're volume overloaded because they've been getting hit with tons of fluid boluses for hypotension in the hospital, whatever it may be, you're trying to get some of that fluid off. These are very good drugs. What we have to quickly talk about though is the concept of the kind of pharmacodynamic, pharmacokinetic aspects of these drugs. So whenever you give these drugs, okay, one of the interesting things about them is that they have a very specific dose-response relationship. So here we have a curve. You thought this wasn't coming back. I know. It's a pain in the nut, but we're going to talk about it. So here we have the dose that you're going to give of the drug on the x-axis, and then the response on the y-axis. When you give this drug, let's say that you give kind of a low dose and it doesn't give you the good kind of response that you want. It's not kind of going up. But then you start to increase the dose, and then you start seeing an increase in the response. And then as you increase the dose, you still see an increase in response, and you increase the dose and you increase the response. But then eventually you keep increasing the dose and you see no change in the response. This is a very interesting thing that you can see with all of these diuretics. The dose that you gave that opened up the gates to produce urine is right here where we start to see this uprise. This is your threshold dose. This is your threshold dose. So this is basically the dose that you give that will really allow for them to be able to produce some adequate amount of urine. And then as you increase the dose, you still get a good response, but then once you hit the maximum dose, you give them let's say that at here, I'm just giving you an example. You want to cause a patient to diurese, and so maybe their threshold dosage is 40 milligrams. I'm just giving this as an example. And you increase it to 60, to 80, to 100, to 120. And then at 120 milligrams, you get no change in the amount of urine that they produced. So at 120 milligrams, or 100 milligrams, so let's say that this point here where it stops kind of increasing the response. Let's say here, this is your ceiling. This is your ceiling dose. Meaning that, let's give an example that this is 120 milligrams. So anywhere from 40 milligrams, once you start that, the patient's producing an adequate amount of urine. If you increase to 60, they increase their urinary output even more. If you increase to 80, they increase. If you increase to 100, they keep putting out more urine. But once you hit at 120 milligrams, the amount that you actually produce is the maximum amount of urine that they will produce. It doesn't matter if you go to 140, 160, 180, 200 milligrams. They will not produce any more volume of urine that they produced at 120 milligrams. So that's one of the interesting concepts of giving diuretics. What it's basically telling you is, if you give a patient a diuretic to try to make them urinate, and you give them 20 milligrams and they don't produce an adequate response, don't give up. That might not be their threshold dose. Double the dose. Give them 40 milligrams. See if they produce an adequate amount of urine. If they do, then they had a response to that. So maybe their threshold dose is above 20. It's 40 milligrams. But if you want them to have them make more urine, they're not making an adequate amount, double the dose again, give 80 milligrams, and they start producing more urine. That's a good response. But you're still not kind of getting them diuresed enough. Double the dose again. If you get to a point where you're doubling the dose and the amount of urine that you're producing is the same at that double dose as it was from the prior, then you're at the ceiling dose at that prior dose. And there's no need to keep increasing the dose. You're not going to get more diuretic effect. That's the concept that I hope makes sense with diuretics. The next concept here is that whenever you think about each one of these diuretics, they have different levels of potency, I think is the best way to saying it. Their efficacy has been shown to be relatively the same, but it's the potency of the drugs that's a little bit different. So let's actually use these in different kind of curves here. So let's say here we have one curve, and we're going to start this one here that it only requires a low dose to get to its maximum kind of efficacy. And then we'll go to this next one here where this one you need a little bit of a higher dose to produce its maximum efficacy. And then here we'll have another one which you require a decently high dose to produce its maximum efficacy. So what you'll notice is, as you kind of go this way, the potency is starting to decrease. So as you move this way, you're requiring a higher dose as you go this way, which means it's going to have a lower potency, right? But they all have the same Emax. So their efficacy is all the same. What is important to remember about this is that these are the most commonly utilized loop diuretics, furosemide, torsemide, and bumetanide. And the most potent one that you can give at a very low dose to produce its maximum efficacy is going to be this red one here. This is going to be, we're going to just use the first letter here, bumetanide. Then the next one, which you can give it a lower dose and still produce a maximum efficacy, is going to be torsemide. And then the one that you can give at a decently high dose to produce the Emax, the same Emax effects as these, is going to be furosemide. They have the same efficacy, but you just have to find the equivalent dose that produces that max efficacy. So that begs the question, what is the equivalent dose that I need to be able to think about whenever a patient is on a particular diuretic? Let's say that I just pick a dosage. This is the way that I learned it, is that you can kind of pick 40 milligrams of furosemide is equivalent to approximately 20 milligrams of torsemide, which is equivalent to approximately 1 milligram of bumetanide. So when you think about that, that's absolutely insane. So if, for example, I prescribed a patient 2 milligrams of bumetanide for fluid diuresis, that's going to be how much Lasix? That's a ton of Lasix, right? So you give 2 milligrams of bumetanide, that's about how much of this? That's an insane amount of furosemide. So when you think about that, it's an important thing to remember that furosemide is going to, in comparison to bumetanide is 40 times more powerful than furosemide. And then torsemide is 2 times more powerful than furosemide. So that's an important concept to really understand when you're thinking about the equivalency between these two drugs. So that's why, again, when we use this graph here, we can utilize lower dosages of this particular drug to produce the maximum efficacy. Lower doses of this one in comparison to furosemide to produce its maximum efficacy. And you have to give a little bit of a higher dosage, again, of furosemide to be able to produce its maximum efficacy. One additional concept here is with respect to bioavailability. Furosemide, the bioavailability that we'll represent with F for these is about 50% for furosemide, and it's generally like about greater than 80%. So almost close to 100% for bumetanide and torsemide. So basically when you give this kind of PO, you got to really realize that whenever you give this via a PO version, you have to half the dose. So giving 40 milligrams of furosemide IV, you'd have to give 80 milligrams of it in a PO form to be able to get that dose. So remember, it's just half. The PO form would be 2 times in this situation. The PO form would be 2 times the IV dose. Okay, so that's important to remember. Because again, if I gave 40 milligrams of furosemide PO, I'm only going to absorb 20 milligrams of it. Whereas if I give the torsemide or the bumetanide, it's pretty much almost approximately equal to the IV dose. It's very, very similar, okay? So that's an important thing to be able to remember when you think about these drugs with respect to kind of their pharmacokinetics and dynamics. All right, so we know now that whenever you give a diuretic, again, you have to give a specific dose to be able to allow for them to diurese. As you go up, you may continue to get response, but once you increase the dose to a point where the response is the same, so in other words, I gave 80 milligrams and they produced 400 cc's of urine, I give 100 milligrams, they produce 400 cc's of urine. The ceiling dose was 80 milligrams. I don't need to give any more because I'm not going to get any more of a response. And then again, on top of that, noting the potency of these drugs is very important. That bumetanide is the most powerful, then torsemide, then furosemide. So I can give lower doses of bumetanide, then torsemide, then furosemide. Okay, and then again, remember that the equivalencies is 40, 20, 1. And the bioavailability of torsemide and bumetanide is almost close to 100%, so the PO to IV kind of conversion is almost exactly equal. But whenever you give furosemide, it's about 50% bioavailable. So again, if you want to give the PO version, you have to be able to give them 2 times whatever the IV dose is, because that's an important thing to remember, okay? All right, my friends. That's the big thing for these drugs with respect to pharmacokinetics and dynamics. Now, obviously, we know that the main indication of these drugs is diuresis. We'll talk about that in a little bit, I promise. Another indication for these drugs besides causing fluid diuresis is they can excrete particular types of molecules. So you know whenever patients have what's called hyperkalemia, so they have high potassium levels within the blood? So whenever patients have high potassium levels within the blood, you can try to treat hyperkalemia a couple different ways. One is you can try to shift it into the cells. One is you can try to poop it out. And then the other way is you can try to urinate it out. And so what we can do is we can give furosemide, because if the potassium is kind of taken into the urine and it's excreted, generally what actually happens is when you give any kind of loop diuretic, they're going to inhibit this sodium-potassium 2-chloride cotransporter. So you reduce the reabsorption of sodium and chloride. So what happens is you get more sodium and more chloride that reaches the distal convoluted tubule, particularly sodium. And whenever you get to the distal convoluted tubule, you have more sodium to reabsorb across the distal convoluted tubule. Now, whenever positive ions are moving out of the tubular lumen into the tubular cell, you're losing positive ions in the tubular lumen. So you need to replace that. So guess who moves across to be able to allow for the sodium to come in? Potassium. So potassium is actually going to be lost in the urine a lot with loop diuretics. So potassium will cause a lot of kaliuresis, potassium excretion. Okay? They do that because they inhibit the sodium reabsorption in the ascending limb. That means a lot of sodium makes it to the distal convoluted tubule. A lot of sodium is reabsorbed in the principal cells, and a lot of potassium is essentially excreted out of the principal cells, and a lot of potassium is lost into the urine. So it treats hyperkalemia by causing potassium excretion. Okay. The other concept, but we don't utilize it as much anymore, is that you can also be utilized to excrete calcium. Again, so when patients have hypercalcemia, again, calcium is usually going to be kind of filtered across, move down the descending limb, move up the ascending limb. And again, whenever you give a loop diuretic, you inhibit these transporters, which then causes less sodium, chloride to be reabsorbed, less potassium to be excreted into the tubular lumen, and that makes the inside of the cell, tubular lumen, more negative, which keeps calcium and keeps magnesium in the tubular lumen and causes calcium and magnesium to be lost. So you're going to have a lot of the calcium that remains in the tubular lumen and then a lot of calcium that gets peed out. So this will effectively cause a lot of calcium to be excreted into the particular amounts of urine. So this can also treat hypercalcemia. However, we don't find this to be as commonly utilized as much as it used to be in the past. but this is definitely a very common indication of giving loop diuretics is to cause potassium excretion, not as much so as in the recent years of giving calcium for calcium, hypercalcemia causing calcium excretion. But now that we understand the indication of loop diuretics, some of the indications, which is again hyperkalemia, hypercalcemia, causing them to become lost into the urine. We understand the pharmacokinetics and pharmacodynamics. We're going to talk about the most common indication of loop diuretics, which is fluid diuresis, but we're going to do it in combination with other diuretics. I promise it'll make sense when we get there. Until then, just trust me. Let's move on to thiazide diuretics, talk about those a little bit, and talk about some of the other kind of miscellaneous indications for that drug category. All right, my friends, so now let's move on to thiazide diuretics. So thiazide diuretics, there's a bunch of different drugs in this category as well. So a couple of them that I want you guys to remember for these ones is the most commonly utilized one is going to be hydrochlorothiazide. So we commonly utilize this as kind of an HCTZ. Okay, so that's one. Another one is called chlorothiazide. So chlorothiazide. Another one is called metolazone. And then another one is called chlorthalidone. And there's another one, there's another additional one called indapamide as well. But these are your thiazide diuretics. There's a lot of these. And they're very commonly utilized. I like to utilize these a lot. But what I want you to remember is, again, thiazide diuretics do what? They block the sodium chloride cotransporter in the early distal convoluted tubule. That leads to sodium, chloride, and water loss into the urine, and they don't actually cause calcium excretion. They actually retain calcium, so they can actually cause hypercalcemia. So there's another indication for this. We're going to talk about the effect of the fluid diuresis from this drug a little bit later. But what I want you to understand is there's other additional indications of this drug. Believe it or not, it actually can cause vasodilation. How it does it, I have no stinking idea. I tried to look for the evidence of this, but I don't know exactly how. But if we have our arterial system, if you think about this, it's kind of two kind of benefits to this. If we take the arterial smooth muscle cell and we give a thiazide diuretic, it may work by some particular action to be able to cause smooth muscle relaxation. So if it causes smooth muscle cell relaxation, this will cause what type of effect? Vasodilation. If you vasodilate, what do you do to your systemic vascular resistance within the arterioles? You would cause a drop in the systemic vascular resistance, which does what to your blood pressure? This will drop your blood pressure. This will drop your blood pressure. So there's one particular interesting thing that you can see from the arterial side with these like thiazide diuretics. So again, if I were to put here hydrochlorothiazide as this drug, it may induce a smooth muscle cell relaxation, which may cause vasodilation. That vasodilation may then cause a reduction in systemic vascular resistance and reduce the BP. Okay, well, that's one interesting concept. The other thing here is that if I block the sodium chloride cotransporter, I block in the early distal convoluted tubule, I decrease sodium reabsorption into the blood, and then subsequently I decrease water reabsorption. So there's less water within the blood. That means I have less blood volume. What does this effectively mean? I have a decrease in blood volume. That means that I don't have as much fluid to bring to the heart. So I effectively reduce the preload. If I reduce the preload, okay, again to the left side of the heart as well, I have less preload. That means I have less stroke volume, less cardiac output, and then effectively less blood pressure, right? So if I drop my blood volume, I drop my end-diastolic volume, I drop my stroke volume, I drop my cardiac output, and I drop my blood pressure. So you're seeing kind of an effect here that I can reduce blood pressure with thiazide diuretics. What would that be good for? Hypertension. So that's one particular indication for thiazide diuretics, is it can be utilized in what's called essential hypertension. Pretty cool concept, right? Especially chlorothiazide, chlorthalidone, and hydrochlorothiazide. Metolazone, not as much, but again, these are good drugs at treating essential hypertension. Because again, they can cause arterial vasodilation, and they can actually cause reduction in, a mild to modest reduction in blood volume, which can reduce preload, reduce stroke volume, reduce what? Cardiac output, and then reduce blood pressure. All right, cool. Another cool concept here is that I told you that these drugs also, again, block sodium, chloride, and water reabsorption, right? Because they're blocking that sodium chloride co-transporter. So you're going to block the reabsorption of these particular substances. But it was mainly the sodium that was really interesting. If we block the sodium reabsorption, it led to less sodium inside of the cell. That means that more sodium could come in the cell and more calcium could be reabsorbed. So the other effect that we talked about when we talked about the mechanism of these drugs is that we also increased calcium. So effectively, when you give these particular drugs, you're inhibiting this transporter here, and the effect is that you're decreasing the sodium chloride and water reabsorption, but you're increasing calcium reabsorption. So that means that less calcium is going to be present in the urine. Why would that be beneficial? You know, in patients who have kidney stones, because they form a lot of calcium, kind of like oxalate or calcium phosphate stones, may be beneficial. So it could be beneficial in patients who have what's called calciuria. Sometimes this is like a familial kind of process, but it increases the risk of renal stones. So if I can give them this drug, like a thiazide diuretics, it may decrease the calciuria and decrease the incidence of nephrolithiasis. That's a pretty cool concept. The other thing is if I actually cause less calcium to be lost in the urine and more calcium in the blood, I may have more calcium for my osteoblasts to deposit calcium into the bone tissue. That may be beneficial in patients whose bone tissue is very porous and weak and very fragile, such as in osteoporosis. So it's important to remember that there is, again, 3 additional indications to thiazide diuretics besides fluid diuresis. They can treat hypertension, essential hypertension, by causing vasodilation of the arterioles and reduced blood volume, which causes reduced preload, reduced stroke volume, reduced cardiac output, reduced blood pressure. They also cause reabsorption of calcium into the blood and cause less calcium excretion, which may be beneficial in patients who have lots of calcium in their urine that increase the risk of kidney stones. So it reduces nephrolithiasis. Plus, in patients who you need to have more calcium for the osteoblasts to deposit calcium into the bone, such as an osteoporosis, this may be beneficial. So that is the concept of these drugs. Again, I want to stress here that I will cover the effect that they have on removing fluid from the body in patients who have CHF, chronic kidney disease, fluid overload states. We will get to that. But for right now, know the extra indications and the names of these drugs. All right, let's move on to the next category of drugs, which is your potassium-sparing diuretics. All right, my friends, let's now talk about potassium-sparing diuretics. So these are the puppies that are working at the later part of the distal convoluted tubule. They're either blocking the aldosterone, which is what kind of drugs in that category? Spironolactone, eplerenone. These are some of the drugs in this category. Spironolactone, lactone, and then another one is called eplerenone. And then another one is the ENaC blockers. So these were blocking aldosterone, which were reducing the sodium potassium pump formation. This was reducing the ENaC formation. This was reducing the potassium channel formation on the apical membrane. This is blocking the ENaC channels directly, which indirectly leads to less potassium moving into the cell, less sodium potassium ATPase activity, and then less potassium being excreted via the luminal channel. And this was going to be amiloride. And then I am not a big fan of this drug, really should stay away from this drug. This is called triamterene. But these are going to be your potassium-sparing diuretics. Now again, we'll talk about how these are utilized in fluid diuresis. We'll talk about how carbonic anhydrases are used, and inhibitors. We'll talk about how loop diuretics are used. We'll talk about how thiazide diuretics are used. And we'll talk about how potassium-sparing diuretics are used in fluid diuresis in like CHF, CKD, acute kidney injury, nephrotic syndrome, fluid overload states, etc. But let's talk about some of the other accessory or kind of additional indications of these drugs. So we talked about how loop diuretics are used in hyperkalemia, hypercalcemia. We talked about how thiazide diuretics are used in hypertension, and then also causing less calcium to be excreted in the urine, which can lead to less kidney stones. And also, it can also help to benefit patients who have osteoporosis. With potassium-sparing diuretics, especially the aldosterone blockers, these are really cool. So what you can see here is that, what if the patient has a disease state? And in that disease state, they have a tumor or some type of process here called Conn syndrome. You know, this is called Conn syndrome. And this causes them to produce a massive amount of aldosterone. So it causes what's called hyperaldosteronism. And whenever they have all this high amounts of aldosterone, what this can actually do is this can work on many different types of intracellular receptors and cause many different types of effects. So hyperaldosteronism, obviously one of the big things that we see is the effect that it can cause a lot of sodium reabsorption, a lot of water reabsorption, a lot of potassium excretion, a lot of metabolic alkalosis. So there's a lot of different things that we could see as an effect of that, right? So some of the big things that you can see with this is called Conn syndrome. It's a syndrome in which there is a massive amount of aldosterone released from the adrenal cortex. And then again, the things that we can see as a result of that is hypernatremia. We can see an increase in water reabsorption, which can cause an increase in blood volume and blood pressure. We can see less potassium present within the blood. We can see a patient developing a very high pH, like a metabolic alkalosis. So this is a lot of different things that you can see as a result of this. What if we gave a particular drug, such as spironolactone or eplerenone, to block the aldosterone, not just in helping to aid in diuresis, but also helping to block the aldosterone side effects that's coming from hyperaldosteronism. This would be a very beautiful indication to be able to give the aldosterone blockers. So this would be the aldosterone blocker effect. Now, the other interesting thing about this kind of concept here is that with these drugs, you may see some potential benefit in hypertension, very mild, utilized primarily in patients who have CHF. So yes, they can be utilized in hypertension. The primary indication by which they truly would be utilized in patients with hypertension is patients who have chronic hypokalemia and CHF, because these will spare the potassium loss, so they'll help to increase your potassium levels, and also they've been shown to reduce cardiac remodeling in patients who have CHF. So it really can be utilized in hypertension, but again, it's not going to be your first-line agent, more particularly in comorbidities such as patients who have heart failure and hypokalemia. But again, nonetheless, let's talk about another concept here. So here, but we'll add this in here. Not only can it be used to treat Conn syndrome, so that's one indication. A second indication, if you really wanted to add this into your list, is it can be utilized in patients with hypertension plus low potassium plus CHF. This may be beneficial specifically for aldosterone blockers. Not as much amiloride and triamterene. There hasn't been enough evidence to support those. All right, especially in CHF. Another indication for these drugs, specifically the ENaC blockers. So another indication, exactly how they do it, I wish I knew. I wish I understood how exactly they do this. There's some theory that it may modulate the renin-angiotensin-aldosterone system process, but amiloride and triamterene may treat a particular condition here. So the ENaC blockers, they may treat a condition where ADH is actually present. It's being produced. It's acting on the V2 receptors on the kidney tubules, but the effect that it's having on the kidney tubules is diminished. So the kidney tubules aren't having adequate V2 receptors to respond to ADH. So ADH is actually present, but they're not responding to the ADH. And so this disease is called nephrogenic diabetes insipidus. And ENaC blockers may prevent this type of process, or can be utilized to treat patients who have nephrogenic diabetes insipidus. Another drug that you can utilize is the Thiazide Diuretics. They may also be beneficial in nephrogenic diabetes insipidus, but again, not a super well-known fact as to how they do that. All right, but nonetheless, what I really want you to understand about the potassium-sparing diuretics, again, is the names of the drugs, okay? Know that first. Also know some additional accessory actions of them. The aldosterone blockers, very good at treating hyperaldosterone states, duh, right? Especially in Conn syndrome. But they also are very beneficial in reducing mortality, especially in patients who have CHF. It's been shown to reduce mortality. If you have hypokalemia, they'll spare the potassium. And if you have hypertension, they may provide some benefit there. Very specifically, though, this is the aldosterone blockers. And then amiloride or triamterene, the ENaC blockers, they may work in some specific way to treat patients who have nephrogenic diabetes insipidus. Okay, at this point, we have covered all of the diuretics. We've covered their names. We've covered their mechanism of action. We've covered some other additional functions besides fluid diuresis. So again, quick recap, mannitol, urea, we talked about them reducing ICP, reducing intraocular pressure, and again, causing massive diuresis in those situations. Carbonic anhydrase inhibitors treating altitude sickness, high altitude pulmonary edema, and again, reducing intraocular pressure in glaucoma and treating idiopathic intracranial hypertension. We talked about loop diuretics being utilized in hyperkalemia, hypercalcemia, and we talked about thiazide diuretics treating hypertension, especially essential hypertension, and also causing less calcium to be excreted to treat patients who have increased calciuria with renal stone risk, as well as helping to reduce the significance of osteoporosis. Potassium-sparing diuretics, we talked about the aldosterone blockers preventing or helping treat Conn syndrome and treating hypertension as long as they have heart failure and hypokalemia. And then again, the ENaC blockers being beneficial in nephrogenic diabetes insipidus. And if you want to add on there, thiazides can also treat nephrogenic diabetes insipidus. All right, we're on to my favorite part of this lecture. Believe it or not, however deep into this lecture we are at this point, but we're at the most important point that I need you to understand here. When we utilize diuretics to pull fluid off of the body, how do we do this and why do we do this? We have a patient who's coming in or is in the hospital, and they're having evidence of potentially like a significant amount of pulmonary edema, a significant amount of peripheral or generalized edema within their belly or within their legs. We need to be able to try to remove some of that excessive fluid, because if they have a lot of pulmonary edema and pleural effusions, maybe it's causing respiratory distress. Maybe if they have a lot of fluid building up within their abdomen, this is actually causing a lot of congestion within the abdomen, reducing urinary output. If it's kind of causing a lot of swelling and distension of the actual legs, this may cause a lot of pain and discomfort. So in those particular indications, we may want to start trying to pull and remove some of the fluid from their body. This would be an indication for fluid diuresis. Now, why would this potentially happen? So why could someone develop a lot of fluid that can actually accumulate within their lungs, leading to things such as pulmonary edema? So we can have a lot of fluid that kind of develops within the interstitial spaces within the lungs and within the alveoli. Or maybe they also have pleural effusions. So maybe they have a lot of these fluids that are accumulating within their pleural cavity here in situations like this, such as maybe pleural effusions. And these are maybe causing a lot of like respiratory distress for the patients. This is definitely going to be one of the more common indications of why you would want to initiate or elicit fluid diuresis. So maybe these problems are causing some degree of respiratory distress. So you could see pulmonary edema and pleural effusions accumulating. Or maybe they have a lot of like peripheral or pitting edema within their legs. Or sometimes, another thing I can find is that sometimes if patients have a lot of congestion within their actual body, sometimes they can have a lot of fluid that backs up into their abdomen. And sometimes this can cause a lot of like, distension of their abdomen. So sometimes we can see big old bellies and kind of a distension of the abdomen as well. So you may even see some type of hepatomegaly or ascites. So you may see some type of ascites or hepatomegaly. These are a lot of kind of like your central kind of congestion. So either way, you're seeing a lot of these congestive symptoms. The patient's coming in, they're in respiratory distress because they have fulminant pulmonary edema and pleural effusions, or they look like the Michelin Man because they're filled up and they have massive amount of generalized kind of peripheral pitting edema, and they have a lot of fluid within their belly causing hepatomegaly and ascites. In these particular situations, you may want to remove some of the fluid from their body and reduce the amount of distress that they may be having. But the question is why are they developing this pulmonary edema, peripheral edema, pleural effusions, ascites, hepatomegaly? The most common cause for this is going to be heart failure. So if a patient has some type of like dysfunction of their left ventricle, so for example, they have heart failure, whether this is heart failure with a reduced ejection fraction, so they have systolic dysfunction, where they can't contract blood out of their heart, or whether they have diastolic dysfunction, so they have a very thick ventricle and they have impaired filling and high filling pressures. So whether this is a heart failure with a preserved or reduced ejection fraction, they have heart failure, and their problem is they're not getting blood out of their heart, or they're not filling the heart properly. So these are the particular problems, is they're having a difficult time being able to squeeze blood out of the heart, or they're having a hard time being able to fill the heart. If that happens, fluid will back up via the pulmonary circulation and lead to pulmonary edema and pleural effusions. That's a problematic issue. If the patient also develops heart failure and this causes them to back up, and then they end up with kind of like problems of the right heart as well, so now they also have right heart problems, then fluid will back up into the right atrium and then down into the inferior vena cava, which will cause ascites, hepatomegaly, and big old swelling of the legs and arms. And so this can lead to a lot of the generalized peripheral edema. In these particular situations, that would be one problematic issue as to why this would happen. So systolic or diastolic heart failure, okay? Another problem is, what if the patient has what's called acute kidney injury or chronic kidney disease? So what if another potential problem here is that the patient has what's called CKD, like really bad chronic kidney disease or an acute kidney injury? So they're basically not very good at producing urine. So if their problem is they can't adequately produce urine, then all the fluid that they're supposed to be having kind of filtering through them and they're supposed to be making is not happening. So their urine production is poor. And so they're not eliminating fluid from the body. If they're not eliminating fluid from the body, they're increasing the amount of blood volume that the patient has. If they're increasing their blood volume, then they're increasing the filling of the heart. And what if the heart already has some degree of systolic dysfunction or diastolic dysfunction? Then it's going to worsen their pulmonary edema. Or if they don't have any problem or dysfunction, but you're constantly filling the heart and congesting it with fluid because you have so much blood volume, this is again going to cause problems in this area and cause pulmonary edema or generalized peripheral kind of pitting edema and ascites and hepatomegaly. Another problem here is maybe it's not due to a chronic kidney disease or acute kidney injury, it's not due to heart failure, systolic or diastolic. Maybe it's because the patient's been in the hospital for a couple days and they've been crushed with like 20 liters of fluid. And they've been giving this patient lots of fluid because maybe they're not making adequate amount of urine, or maybe they're hypotensive, or maybe they appear dehydrated, whatever it may be. That could be another very, very common issue. I can't stress how much I've seen this all the time. But fluid overload states and situations where it's iatrogenic, so massive amounts of IV fluids for the patient, can also cause this problem where you're just kind of crushing them with large amounts of fluid. And if you're just pushing fluids into their vascular spaces, all you're doing is you're massively increasing their blood volume. And if you're increasing their blood volume, you're increasing the filling of their heart. If you're increasing the filling of their heart, you may cause a lot of congestion. If they don't have any underlying heart disease, it may not be as bad. But if you're crushing them with tons and tons and tons of fluid, that preload may be too much. It may cause excessive congestion of the heart and then decompress back into the lungs or decompress into your actual peripheral systemic circulation, such as in the legs, the belly, or in the arms, and cause a lot of like generalized peripheral pitting edema. All right, so again, 3 particular problems as to why I see patients developing a lot of like edema symptoms is due to congestive heart failure, most common, chronic kidney disease or acute kidney injury where they're not making an adequate amount of urine and becoming fluid overloaded because they can't eliminate fluid from their body, or they're getting a lot of fluid iatrogenically from intravenous fluid boluses or infusions, and it's causing fluid overload states as well. Again, if you have CKD, acute kidney injury, fluid overload states, and have heart failure, it definitely will exacerbate this situation. But you can have all of these symptoms with just fluid overload or just CKD without any heart dysfunction, just realizing that. But nonetheless, patient has massive amount of pulmonary peripheral edema. They come to you, all right, you're taking care of them. They're, let's say, just to give you an example, let's say that you're looking at them and they have pulmonary peripheral edema and also their weight is increased by, let's say, 10 kilograms. I'm just making a massive amount here. And let's say that their I&O balance, so the amount of fluid that they're getting in versus out for stay, so they've been in the hospital, is let's say that they're 10 liters positive at this point. I need to try to get that fluid off. I need to get rid of the fluid in the lungs, get rid of the fluid in the pleural cavity, get rid of the fluid in the abdomen, get rid of the fluid in some of the actual interstitial spaces in the legs and the arms. How am I going to do that? Great question. Here's what I'm going to do. I'm going to start with the most powerful diuretic, which is going to remove the largest amounts of volume from the body. Because again, what I'm trying to do when I give diuretics is I'm trying to have them do what? I'm trying to have them do what to the urine? Increase the sodium in the urine, increase the water within the urine, maybe increase some of the chloride in the urine, but get rid of a lot of these particular things. Because if you get rid of these, then what happens is you have less water, less sodium in the blood. That means you have less blood volume. That means you have less preload. And that means that if you have less preload, you're going to have less congestion of the heart, and you're not going to keep kind of decompressing the heart backwards into the pulmonary or peripheral systemic circulations. So that's the whole concept here. So in order for me to do that, I want to work at blocking specific points. What's the most powerful one that I'm going to start with? The most powerful one that I have to start with my regimen here is I'm going to start with a loop diuretic. A loop diuretic should be first within this kind of regimen. Why? Because it's the most powerful and it's going to remove the largest amounts of sodium and water. So I'll give a loop diuretic. And what is it going to block? Let's see if you guys remember. It's going to block the sodium, potassium, 2-chloride cotransporter, which is going to block sodium chloride reabsorption as well as subsequently water reabsorption. So what will end up in the urine, I get rid of a lot of sodium, I'll get rid of a lot of water, and I'll get rid of some chloride. And additionally, I'll get rid of calcium, magnesium, some other things like that, right? But I'm going to really kind of decompress and get rid of a lot of blood volume. So that would be my first part of the regimen. Now, as I do the loop diuretic, again, take into consideration the threshold and ceiling doses. So maybe you give them a particular amount of the drug, it's not enough. Maybe you have to increase the dose until you get an adequate response. But once you hit a particular dose where I give them 80 milligrams and they produce 400 cc's of urine in 2 hours, and I give them 100 milligrams and they produce the same amount, the ceiling dose was 80. I don't need to give any more. So remember that concept. But I've given them this and they're still not producing an adequate amount of urine, because they're still not kind of getting that balance down. What I'll do is I'll consider a second drug, and there's even a better indication for this. So I'll start off with the loop diuretic, and then what I can do is I can add on a second drug. This is going to be where I add on the Thiazide Diuretics. And there's 2 indications of why I would add on the Thiazide Diuretics. Okay, one is I'm going to increase my diuresis. So one indication is you're going to increase your diuresis. So you're going to augment and enhance the effect of the loop diuretic, because if you give a thiazide, you'll cause more sodium and more water loss, right? Because now I'm going to block the sodium chloride cotransporter here in the early distal convoluted tubule and cause more sodium and water to be lost in the urine. So I'll increase diuresis. Here's one more thing. Believe it or not, with loop diuretics, they cause more water loss than they do sodium loss. Let's write that down. So with loop diuretics, they actually do cause more water loss. So if we actually put like with respect to loss, they actually cause more water loss than sodium loss. So if you cause more water loss from the body than you do sodium loss, what's going to happen to the actual sodium? It may cause hypernatremia. So believe it or not, this is a potential effect here, is that you may see patients develop what's called hypernatremia within the blood. So in that particular situation, guess what thiazide diuretics do? They cause a good amount of sodium excretion. So because of that, they actually cause more sodium excretion than they do water excretion. So when we think about this, they actually, with their loss, they actually cause more sodium excretion than they do water excretion. So what will they do? They'll lower the actual serum sodium within the blood. So if a patient's getting hypernatremic, another indication to give a thiazide diuretic is because they have hypernatremia and I'm going to cause more sodium loss. So I start off with a loop diuretic to pull off fluid. They start to develop hypernatremia, add on a thiazide diuretic. Or they're not getting enough diuresis, add on a thiazide diuretic, okay? As you do this, you start to notice a potential problem that happens. And this is really kind of interesting. When you give loop diuretics and you give thiazide diuretics, they really try to cause a lot of sodium wasting. So you really try to get a lot of sodium in this distal part. So you're going to block sodium here, so you have a lot of sodium that moves from this point. You're going to block sodium here, so you're going to have a lot of sodium kind of at this point. And whenever the sodium gets to the late part of the distal convoluted tubule, you have a lot of sodium there. When sodium kind of actually gets at this point here, it can actually get kind of absorbed, reabsorbed, because there's lots of sodium in the lumen and less sodium in the cell. So it kind of gets reabsorbed into this distal part. This is the distal convoluted tubule, the distal part here. And what happens is, whenever you kind of cause a lot of this sodium to actually be reabsorbed, there's another channel here. There's a channel here, let's actually do this one in pink here, that actually kind of excretes a molecule in combination with the sodium. When the sodium gets reabsorbed, it kind of causes the positive charges to be lost, because now sodium's moving in, the inside of the actual lumen's getting negative. So it pulls with it protons. So now I start losing lots of protons into the urine. What else did I say happens? As you pull sodium into the cell, the inside of the lumen becomes negative. It pulls protons, but guess what else it pulls? Potassium. And you lose potassium into the urine. So I start to see 2 potential effects as I give loop diuretics and thiazide diuretics. I start to see hypokalemia, and I'm losing protons into the urine. If I lose protons, so then the next concept here is as I actually give loops and thiazides, I get 2 potential problems that arise. I cause hypokalemia because lots of sodium gets reabsorbed in the distal convoluted tubule, which causes potassium excretion. And then lots of sodium gets reabsorbed in the distal convoluted tubule, which causes proton excretion. So this is going to lead to hypokalemia and low protons within the blood. This will then lead to, if you have low protons in the blood, what's going to do to the pH? It's going to increase the pH. This is called metabolic alkalosis. If only there was a drug that I could give that would actually cause metabolic acidosis. There is. Carbonic anhydrase inhibitors. So now what I'm going to do is I'm going to add on another drug here. So the next drug that I'm going to add on would be my carbonic anhydrase inhibitors. Carbonic anhydrase inhibitors. And why would I add these drugs on? Because they're going to cause a lot of bicarb loss. So these increase your bicarbonate loss, which does what to the pH? What does it do to the pH, my friends? It causes the pH to go down. It causes a metabolic acidosis. This is a good indication because what happens is with a loop diuretic and a thiazide diuretics, they cause metabolic alkalosis. So the carbonic anhydrase inhibitors will combat the metabolic alkalosis caused by the loop diuretics and thiazide diuretics. Why is that a problem? Because whenever the patients start getting metabolic alkalosis, whenever they get metabolic alkalosis, it actually impairs diuresis. So it decreases the efficacy of the loop diuretics and the thiazide diuretics. So remember, as the pH starts getting higher, it actually inhibits the efficacy of loop diuretics and thiazide diuretics. So if I try to decrease the pH and get it to at least normal, then what am I going to do? I'm going to increase the efficacy of my loop diuretics. So there's two concepts with this. As I cause bicarbonate loss, it decreases the pH, which combats metabolic alkalosis and improves or increases your diuresis. Plus, you also get a little bit of sodium blockade, right? So if I block the carbonic anhydrase, if I block this, I'm going to not only block sodium reabsorption, but I'm going to block bicarb reabsorption. I'll block this process here. And so I'll cause bicarb to be lost in the urine and a little bit of sodium and subsequently a little bit of water. So I'm going to get a little bit of an extra diuresis effect and I'm also going to combat metabolic alkalosis. But I have another problem. My loop diuretics and my thiazide diuretics were causing hypokalemia. And now I got to give all this potassium back to the patient because I don't want them to develop an arrhythmia. So what do? I can give them potassium. I can do that. But what if I have another drug that can also increase my diuresis and also increase the potassium and kind of prevent the potassium wasting that the thiazides and loops are causing? What if I had a drug like that? I do. So that's where the fourth drug comes in. The fourth one that you can add on here, you can tell this kind of stuff gets me super excited, it's pretty cool, is going to be your potassium-sparing diuretics. So this is your potassium-sparing diuretics. So this could be the aldosterone blockers or this could be the ENaC blockers. But I'm giving these drugs because they're going to increase the potassium retention or decrease potassium loss, right? So effectively what they'll do is you'll increase your potassium inside of the blood, which will combat the hypokalemia that the loops and thiazides were causing. And believe it or not, guess what the hypokalemia can do? It can impair the efficacy of loops and thiazides. So if I give them a potassium-sparing diuretic, it'll keep their potassium levels high, which will combat the hypokalemia caused by the loops and the thiazide diuretics. On top of that, they do block the sodium reabsorption and potassium excretion in the late distal convoluted tubule and lead to a little bit of sodium and water loss. So you will get a little bit of an increased diuresis effect. So this is my perfect kind of like diuretic regimen. When I have a patient who's extremely in distress from pulmonary edema, a lot of pleural effusions, a lot of generalized systemic pitting edema, peripheral edema, a lot of ascites, hepatomegaly due to heart failure, CKD, acute kidney injury, fluid volume overload. In these states, you need to diurese them. Start with a loop. They may get hypernatremia because it causes more water than sodium loss. If you need to increase your diuretic effect and prevent hypernatremia, give a thiazide diuretic. As you start to treat them with that, they start to get hypokalemic and metabolic alkalosis develops. That impairs their efficacy of those drugs. Give a carbonic anhydrase inhibitors to cause bicarb loss and decrease the pH to combat the metabolic alkalosis from loops and thiazides. And it enhances a little bit of sodium and water loss. If they become hypokalemic, guess what else you can do? And hypokalemia can impair the efficacy of loops and thiazides. You can give a potassium-sparing diuretic like spironolactone, eplerenone, amiloride, triamterene. And what will they do? They will cause potassium retention. They'll inhibit potassium excretion, cause the potassium levels to go up. And if the potassium levels go up, that combats the hypokalemia that the loops and thiazides cause. On top of that, it also blocks sodium and water reabsorption in the distal convoluted tubule and causes a little bit of that to be lost in the urine. So it also causes diuresis. This is the perfect schematic for a patient who has the need to be diuresed. There's one more indication for diuresis, which is a little bit odd and wonky, and that's when patients have a lot of ascites and kind of hepatomegaly and peripheral kind of a pitting edema, but it's due to cirrhosis. In that situation, the treatment's a little bit different. Let's talk about that now. All right, my friends, so now what we're gonna do is we're gonna talk about how do we diurese patients that actually have ascites. You would think, oh, it's the same thing, Zach. You just give them, furosemide and give them like thiazide. Believe it or not, it's not exactly the same. It's a little bit different. And the reason why is the pathophysiology slightly changes. So when patients develop a lot of edema, such as, for example, maybe it's not just ascites, maybe they also develop, a lot of like leg edema and stuff like that too. So maybe they develop a lot of like pitting edema, okay, within the legs, or maybe they develop a ton of ascites, okay, because of a lot of fluid backing up from their liver into the actual abdomen. So in these particular situations, the primary etiology of this is not a fluid overload state, it's not a chronic kidney disease state, it's not a heart failure type of state. In these situations, the primary etiology here is that they have cirrhosis. So there's some type of cirrhosis type of process. And with cirrhosis, their liver becomes so fibrotic that what it does is it actually kind of causes the portal vessels that are supposed to enter into the liver and bring fluid kind of through it, generally blood through it, and then its liver's supposed to kind of remove some particular toxins, remove some particular nutrients, store some particular things, and then secrete some things, and then put it into the inferior vena cava. That's the whole job of it. Problem is that all this cirrhosis and fibrosis starts kind of encroaching on some of these portal veins. And then you start getting a lot of this fibrosis that kind of encroaches onto the portal veins and causes the pressure, because it starts squeezing on it. It starts causing the pressure of the portal circulation to increase. So subsequently what happens is, with the cirrhosis, it causes an increase in the portal blood pressure. Now, with that being said, what happens is the liver says, okay, hold my beer, I got a way to fix this. And it says, I'm gonna release particular types of peptides like nitric oxide and things of that effect, which are going to kind of try to cause vasodilation of this portal vein to try to help to be able to increase the blood flow. Because that's the whole problem. The blood flow is being affected because I have all this fibrotic tissue around it that's compressing the portal vein. So maybe if I try to make the portal vein dilate a little bit, it'll reduce the pressure and help to maybe improve some of the flow through the liver. The downside of this is something else. When you release nitric oxide, what it does is not only does it try to cause a vasodilation effect of the portal veins, it also vasodilates a ton of like splanchnic vessels. Like it really vasodilates a lot of the vessels inside of the abdomen. And the downside of that is that if you really kind of like, let's say that here, I have some particular vessels here. So this one is supposed to be going to the kidney, right? But I have all these other blood vessels that are supposed to be going to other particular tissues. So maybe I have some blood that's going to be going to a bunch of different organs here. So let's say that we kind of put some organs here. So a lot of my GI organs here. And then this one is supposed to be going to the kidney. All right, so this is the blood flow that's supposed to be going to the kidney. What I'm going to do is I'm going to get these nitric oxide molecules and they're going to vasodilate these splanchnic vessels. So they're going to vasodilate. It's going to cause splanchnic vasodilation, which will help to increase the blood flow to these actual organs. But what it'll do is it'll reduce the amount of perfusion to the kidneys, right? Because you're taking and kind of diverting the blood volume away from the kidney and then instead going to the organs. The downside of that is that you have effectively out of this, because you're not going to get as much perfusion to the kidney, you're going to have a reduced renal perfusion. So the actual renal blood flow is decreased. When you decrease the renal blood flow, what this does is this activates what's called the renin-angiotensin-aldosterone system. So then from here, my kidneys are going to start pumping out renin, which will eventually lead to, I'm going to kind of divert throughout the entire process here. It's going to go to angiotensin 2 eventually. Angiotensin 2 is then going to try to go and kind of like squeeze your blood vessels and increase your blood pressure and try to perfuse the kidneys. It's trying to fix that problem. But it also activates aldosterone. When it activates the adrenal cortex to make aldosterone, aldosterone will then go to the actual distal convoluted tubule. And what it's going to try to do in the distal convoluted tubule is reabsorb a lot of sodium, excrete potassium, and then if you also have ADH present, not only will it reabsorb sodium, but it'll also reabsorb water. So you're trying to increase your sodium reabsorption and increase your water reabsorption. The problem with that is that if you kind of have from this effect an increase in sodium reabsorption and an increase in water reabsorption, guess what that's going to propagate? It's going to propagate worsening ascites, and it's going to propagate worsening. So the problem with this is it's going to cause worsening ascites and worsening pitting edema. Okay, that's the problem with this. So now what I got to do is I got to come up with a drug that'll really help me to reduce, in some degree, this. This seems to be the problem here. This really seems to be the problem, is that whenever the liver has cirrhosis, portal blood pressures rise, release vasoactive peptides, it causes splanchnic vasodilation. So it increases the blood flow and diverts it to the actual GI organs, but less of it goes to the kidneys. Less renal blood flow activates the renin-angiotensin aldosterone system. Aldosterone causes a lot of sodium retention, a lot of water retention. A lot of sodium and water gets actually kind of retained within the belly and within the legs. And then before you know it, they're walking around with this kind of like fluid-filled kind of like cavity here in their belly and within their legs, propagating worsening ascites. So what if I gave a drug that blocked aldosterone, which reduced sodium and water retention, which reduced this ascites process? Guess what we got? Aldosterone blockers. So that is where the primary drug for treating patients who have edema, such as ascites, hepatomegaly, peripheral pitting edema from cirrhosis, is actually going to be aldosterone blockers. Because if I give an aldosterone blocker such as spironolactone or eplerenone, I'm going to block the effect of aldosterone on the distal convoluted tubule. I will not reabsorb sodium, I will not reabsorb as much water, and I will not excrete as much potassium. So then effectively my kidneys won't retain as much sodium, I won't retain as much water, and I won't actually have as much of this ascites, and I won't have as much of this pitting edema. Isn't that cool? So this is actually the first line drug, and you actually give pretty high doses of them. So generally very, very high doses of these drugs in patients who have ascites, hepatomegaly, peripheral and pitting edema from cirrhosis. The drug that you can add on as an additional kind of like supportive therapy here to reduce sodium and water retention, which one causes the maximum amount of sodium and water loss? Out of all the diuretics, which one causes the maximum amount of sodium and water loss? Loop diuretics. So the additional therapy that I could add on here would be my loop diuretics. But these are going to be usually in lower doses. And then again, why is this going to be beneficial? Because with loop diuretics, and this is going to be at lower doses. With these drugs, what they're doing is they're blocking the sodium potassium 2 chloride cotransporter and causing a massive amount of sodium and water to be lost into the urine, which again effectively leads to less sodium and water would actually be retained within the blood, which leads to less kind of like sodium retention and water retention within the belly and the legs, leading to less hepatomegaly, less ascites, and less generalized and peripheral edema. Okay? So again, that seems to be the primary therapy for patients with diuresis, due to acidic fluid accumulation or peripheral pitting edema due to underlying cirrhosis. These patients may also potentially benefit from like a paracentesis. So generally when you have a lot of acidic fluid, they may benefit from kind of tapping into the belly and pulling off some of that fluid, such as a paracentesis. And they also may benefit because in patients who have cirrhosis, they have less albumin production. You may actually benefit from giving these patients albumin as well. But again, nonetheless, what I want you guys to understand is how do you remove that fluid with diuretics it's primarily going to be the aldosterone blockers like spironolactone, and then adding on a loop diuretic to enhance sodium and water excretion, reducing the acidic fluids. All right, my friends, the last category here is we give these drugs. We give this patient a particular loop, thiazide, potassium-sparing, carbonic anhydrase inhibitor, or any of these drugs. What are some of the adverse effects that I have to be aware of, and how do I make it easy in my brain to remember them? I got you. Let's talk about it. All right, my friends, so adverse drug reactions. Let's go through them. Ready? First one, loop diuretics. All right. When we talk about loop diuretics, we talked about how they're going to cause the most amount of sodium and water loss. But what I tell you is I told you that, remember, when we talk about loss, it causes definitely a lot of sodium loss and a lot of water loss. But in comparison, because it's going to cause more likely hypernatremia. Why? Just again, as a reminder, yes, you lose a lot of sodium. 25% of sodium is actually going to be blocked from being reabsorbed at the ascending limb, but you'll also lose a lot of water. So because of that, water loss is going to be greater than the sodium loss. And that's why you'll see hypernatremia potentially with loop diuretics. The other thing is that it can actually kind of pull off a lot of water to the point where it can actually cause them to become hypovolemic. So watch out, whenever patients sometimes become over-diuresed, you want to watch out for any types of hypovolemia. So watch out for any low blood volume that can actually cause low blood pressure. So be careful with that. The third thing here, so again, we're really kind of inhibiting this particular transporter, the sodium potassium 2-glycotransporters. That blocks sodium reabsorption, that blocks water reabsorption. We talked about if it blocks sodium and water reabsorption, you're going to lose a lot of blood volume, right? It also can cause more water loss than sodium loss, so you may see hypernatremia, especially with repeated dosing. The next thing is when you block the sodium potassium 2-glycotransporters, it also blocked what else from being reabsorbed in between the cells? You guys remember? Calcium and magnesium. So you should also see low calcium levels in the blood, and it also should cause low magnesium levels within the blood. What was another concept here? Another one with electrolytes is that because you inhibited a lot of sodium reabsorption, a lot of sodium makes it to this distal convoluted tubule. So you're going to have a ton of sodium here. Remember I told you that in the principal cells, you'll reabsorb a lot of sodium into the principal cells, and you'll reabsorb a lot of sodium into these intercalated cells. I told you I'd talk about these eventually. The intercalated cells are the ones that control acid-base balance. The principal cells are the ones that control your electrolyte balance, like sodium, potassium. So remember, sodium, if there's a lot of sodium that gets here, because you blocked it from being reabsorbed, a lot of sodium will move in via this sodium transporter, and a lot of potassium will be excreted into the urine. A lot of potassium. On top of that, if you have a lot of sodium being reabsorbed, it also causes a lot of protons to be excreted into the urine. And again, why does this happen again? The whole concept is as you take a cell and you have to have positive ions flowing into it, such as sodium, that means that you're going to have positive ions moving into the cell. That means that the lumen is going to become negative. When the lumen's negative, it will pull out positive ions such as protons and such as potassium ions. And that's why potassium and protons will be lost into the urine. So because of that, if you lose potassium into the urine, it's going to cause hypokalemia. And if you lose protons into the urine, it's going to make the pH, you're going to have less protons within the blood. If you have less protons within the blood, that's actually going to cause the pH to go up. So it's going to cause a metabolic alkalosis. Okay? So far, hypernatremia, hypovolemia, hypocalcemia, hypomagnesemia, hypokalemia, and metabolic alkalosis. What else? This one's interesting. At the proximal convoluted tubule, you also have an organic acid transporter that is supposed to kind of pump furosemide, but it also pumps out what's called uric acid. And so sometimes these can compete. And so you can't excrete the uric acid as much because you're trying to pump furosemide in there. Sometimes you can't pump out the uric acid. And so uric acid levels actually get accumulating within the blood because you can't pump out the uric acid at the same time you're trying to pump out the furosemide or any of the loop diuretics. And so because of that, the uric acid levels increase in the blood, and that causes hyperuricemia. Okay, another effect. It's weird, but loop diuretics may have some effect on insulin, whether it's inhibiting insulin secretion or whether it's inhibiting the effect that insulin has on tissue cells to bring glucose into the cells. So whether loop diuretics inhibit insulin release or inhibit the effect of insulin on the tissue cells, you're not going to get glucose coming into the cell. And so what happens is the blood glucose levels rise. And so this may also cause the patients to develop hyperglycemia. Hyperglycemia, so increased glucose levels within the blood. There's one more effect, and this is usually at very, very high doses. It may also cause ototoxicity. So toxicity to the actual inner ear and cause hearing loss. Usually that's with ethacrynic acid or super, super high doses of loop diuretics. Okay, but more common with ethacrynic acid. Okay, the next one, let's actually move on to the thiazide diuretics. And the reason why I want to do that is because they have a lot of the similar types of effects. Again, when we talk about this, what will it do to the sodium? Well, I told you that with this, it's actually blocking the sodium chloride cotransporter. And it's going to cause a lot of sodium loss in the urine, but more powerfully, water loss. So what would I see more of an effect of? Hyponatremia. So I'm going to see low sodium in the blood because it's actually going to cause the sodium loss to be greater than the amount of water that is lost. Okay, beautiful. The next concept here, let's actually make this pretty. This was supposed to be a red parentheses. Come on, man. And then we go here, same thing. Low sodium because you're causing more potentially sodium loss than you have water loss. This won't cause as much water loss though, so you won't get as much of a hypovolemia type of effect. So that's a cool thing. The second thing, what can it do to calcium? Oh, you guys better not forget this. If I inhibit the sodium chloride cotransporters, I inhibited sodium reabsorption, which also led to more sodium coming into the cell this way and more calcium coming out this way. And so that causes a lot more calcium to be absorbed. So what will this do to the calcium? It'll increase the calcium levels. So it may cause hypercalcemia. It does cause a little bit of mag to be lost. I'm not going to go over the mechanism here. It's a little bit interesting, but it can cause a little bit of hypomagnesemia. What about the potassium? Well, the same thing. It's blocking sodium reabsorption here in the early part of the distal convoluted tubule. So by the time it gets to the late part of the distal convoluted tubule, you're going to have a lot more sodium than you usually would. So that means a lot more sodium enters into the principal cells, a lot more sodium enters into the intercalated cells. That makes the inside of the lumen negative. That draws out potassium and draws out protons. And so what do I urinate a lot of into the urine? I have lots of protons in my urine and lots of potassium ions into the urine. So effectively this will cause hypokalemia. It'll also cause low amounts of protons in the blood, which will cause an increase in the pH, causing metabolic alkalosis. Okay, what else can it do? The other thing is that, you know, thiazide diuretics are also excreted or pushed into the tubular cells via the organic acid transporter in the proximal convoluted tubule, just like loop diuretics. But guess who else is supposed to be excreted that pathway? Uric acid. And so because uric acid has to compete with both loop diuretics or thiazide diuretics, you're not going to be able to excrete it because you're supposed to be able to excrete thiazides via this transporter and loop diuretics, and now you can't excrete uric acid, and so it builds up in the blood. And so it may cause hyperuricemia. Oh my gosh, man, we're getting good. The other concept here is that it may, again, do something with insulin. Whether or not it actually may inhibit insulin production or inhibit the effect of insulin has on the tissue cells to shift glucose in there, it may in some way either inhibit insulin production or inhibit the sensitivity of insulin on the tissue cells, and so they don't reabsorb glucose or absorb glucose. And so what will this do? This will cause hyperglycemia. It has no effect on ototoxicity. And then one other effect that you actually can potentially consider here is through mechanisms that I'm not completely sure of, it may also increase your lipid levels. So it may also cause hyperlipidemia. All right, so with loop diuretics, you get hypernatremia, hypovolemia, hypocalcemia, hypomagnesemia, hypokalemia, metabolic alkalosis, hyperuricemia, hyperglycemia, ototoxicity. With thiazides, you get hyponatremia, hypercalcemia, hypomagnesemia, hypokalemia, metabolic alkalosis, hyperuricemia, and hyperglycemia and hyperlipidemia. All right. Potassium-sparing diuretics. With potassium-sparing diuretics, it's actually not too bad, thank goodness. With these ones, what you really want to remember here is that what they're doing is, in general, regardless if it's an ENaC blocker or whether it's an aldosterone blocker, we already know that it's preventing sodium reabsorption, which leads to less sodium potassium. So here, really quickly. I don't reabsorb sodium, therefore I can't pump it out via the sodium potassium ATPase. So that means less sodium is being reabsorbed. That means less potassium is being pumped into the cell. That means that there's less potassium to be pumped out of the cell. So that means that less potassium is going to end up into the urine. So because of that, this will cause the potassium level within the blood to increase. So it may cause hyperkalemia. Okay? The other concept here is that this is what's really interesting. With aldosterone has been shown to not only cause stimulation of sodium and reabsorption, potassium excretion, but it's also been shown to stimulate proton excretion into the urine. So if I give an aldosterone blocker, guess what I may potentially do? Inhibit proton excretion. And so another potential effect that I can see here is I'm going to see less proton excretion. So if I see less proton excretion, the protons in the blood may go up, and this may decrease the pH. So this may cause a metabolic acidosis. Another concept here is really cool, is that let's say that we talk about the ENaC blockers. Either way, both of them, both the aldosterone and ENaC blockers lead to hyperkalemia. Why is that important? If the potassium levels in the blood are high, what you guys know is that sometimes we have these transporters on many different cells in the body. And what this does is this transporter is supposed to pump the potassium into the cell and pump the protons out of the cell. Well, the higher the concentration is the potassium within the blood, the more potassium goes in and the more protons goes out. Well, if I have hyperkalemia, I'm going to be shunting in potassium and shunting out protons. So I increase my protons within the blood, which leads to a drop in the pH. So you can see this with both the ENaC blockers and the aldosterone blockers. So I see hyperkalemia. I'm also going to see a metabolic acidosis. There's one more effect, and this is particularly with the aldosterone blockers. Because it not only blocks aldosterone, it can also block some other steroid hormones. Particularly in men, this may lead to the development of excessive breast tissue called gynecomastia. I want you to remember that primarily with the aldosterone blockers. You will not see that with the ENaC blockers, such as amiloride and triamterene. All right, my friends. So with this being said, the primary adverse effects here is going to be hyperkalemia, metabolic acidosis, gynecomastia, or some type of erectile dysfunction or amenorrhea or menstrual irregularities in females due to the aldosterone blockers not only blocking aldosterone, but other steroid hormones. Okay. That's the big things that I want you to remember there. The next one and the last one here is carbonic anhydrase inhibitors. With carbonic anhydrase inhibitors, what we know is they're going to inhibit the carbonic anhydrase enzyme. Therefore, you're not going to be able to reabsorb bicarb and excrete protons. So the proton excretion is decreased, and the bicarbonate reabsorption is decreased. So I lose a lot of bicarb into my urine, and I don't have as much bicarb being reabsorbed into the blood. So my bicarb in the blood is less. If my bicarb in the blood is less, what is that going to do to my pH? It's going to decrease my pH, causing a metabolic acidosis. But I'm going to cause a lot of bicarbonate to end up in my urine. You know what bicarbonate in the urine actually can do? It can increase the risk of kidney stones. It can actually make the urine more alkaline. And if you make the urine more alkaline, it can actually increase the pH of urine and can increase the risk of kidney stones, especially some of the calcium stones. So that's another thing to be able to think about. There's one more thing. If I block the reabsorption of bicarb and I block the excretion of protons, where does that proton problem come into play? Well, you know there's another molecule that's supposed to be kind of excreted from the body that can be very toxic. This is called ammonia. So you can excrete an ammonia, right? Now ammonia, generally what it'll do is it'll combine with this proton. When ammonia binds with a proton, it'll form something called ammonium. And the benefit of this is that when you think about things being absorbed, a thing that's charged is hard to absorb, right? Because you need some type of transporter. Whereas ammonia, this is actually not charged at all. So it could reabsorb right across the actual membrane. So if I weren't having any protons to bind with the ammonia can just get reabsorbed and increase within the blood. Oh my gosh. Hyperammonemia is super dangerous, but that is a potential complication. So watch out for high levels of ammonia within the blood, which can lead to encephalopathy. It can lead to different types of paresthesias and a lot of like cerebral edema types of effects. So again, a big thing to think about with carbonic anhydrase inhibitors is it can cause a metabolic acidosis. It can actually cause the urine to be more alkaline, which increases the risk of kidney stones. And it also can cause hyperammonemia. And then one other thing is that if you do have some degree of sodium reabsorption that's inhibited to this process, you will get a little bit of sodium at the end of the distal convoluted tubule here. And so you have more sodium reabsorption, more potassium excretion. If you really wanted to remember this, this may cause a little bit of hypokalemia as well. My friends, we've covered all the diuretics. I know it's been a beast. I hope that it made sense. I'm so sorry that it went so long, but I hope that you guys did help you and you guys enjoyed it. We got to do a quick couple of cases and then we're done. All right, meet me there. All right, my friends, let's do some cases. So first question here is an elderly patient with a history of heart disease, has difficulty breathing, is diagnosed with acute pulmonary edema. Which treatment is indicated? Well, in general, as we talked about with diuretics, the one that's actually going to be the most effective at pulling out large volumes of kind of fluid from the body, really reducing the true total volume, blood volume within this situation, is going to be loop diuretics. They are going to be the most effective at being able to reduce the actual fluid overload states in the situation of acute pulmonary edema. So furosemide should be the best answer. Acetazolamide is not going to be able to pull that much fluid off. Again, it's good when patients who you're diuresing with furosemide start to become a little bit more metabolic alkalotic, you could add that drug on. Chlorthalidone is a thiazide-like diuretic, so it's going to be more beneficial, kind of like an add-on therapy if your patient's becoming extremely hypernatremic. Again, we also can utilize these in hypertension, and also, again, these drugs may be somewhat beneficial in patients who have some type of problem where their calcium levels within their blood are low, they're osteoporotic, and you want to increase their calcium levels. So those could be indications. And spironolactone is not a super great diuretic for especially acute pulmonary edema. So because of that, I would not be utilizing that drug. So it should be C, furosemide. Altitude sickness. A group of college students is planning a mountain climbing trip to the Andes. What is the most appropriate for them to take to prevent altitude sickness? Well, we already know that it's got to be acetazolamide. So acetazolamide, also known as Diamox, is going to be a nice carbonic anhydrase inhibitor. And so that's really going to help to be able to, again, get rid of a lot of the actual bicarb in the urine, and so that will help to kind of allow for the pH to become a little bit more acidotic. So in these situations here, that is going to be the best particular drug in this situation here would be the acetazolamide, and so that would be C. Alcoholic male has developed hepatic cirrhosis to control the ascites and edema. Which should be prescribed? So this is actually a very interesting one. And so you would think, okay, edema in general, edema is going to be best with things like furosemide. And that would seem to be true in most particular scenarios. However, that's not the most effective drug in classic cases patients who have hepatic cirrhosis. In patients who have hepatic cirrhosis, sometimes they can actually get these weird hepatoadrenal syndromes where they can have these problems with aldosterone levels being a lot higher. And so in those states, we actually have found that spironolactone has actually been the best at treating the hepatic-related edema. So any hepatic-related edema, especially with these hyperaldosterone states, spironolactone is actually shown to be the most effective diuretic as compared to all of them. So spironolactone would be the effective answer here. All right, question 4. 55-year-old male with kidney stones needs a medication to decrease urinary calcium excretion. Which diuretic is the best for this indication? So as we talked about below, we want to know which one helps to be able to increase calcium absorption, basically, is the way that we want to think about this. So if instead of actually allowing for things like a loop diuretic, that's actually going to cause calcium loss. Spironolactone doesn't really have any true effect at all on calcium, triamterene as well. So it leaves us with hydrochlorothiazide, and again, we know that thiazide diuretics help to be able to, again, particularly increase calcium absorption and inhibit sodium chloride and water reabsorption. So this will be beneficial at helping to be able to increase blood calcium levels. Kind of a beneficial situation in patients who actually not only are having problems with kidney stones, but also patients who have osteoporosis. So hydrochlorothiazide would be the choice. 75-year-old woman with hypertension, glaucoma, being treated with chlorthalidone, amlodipine, lisinopril, and acetazolamide. In clinic today, she complains of acute joint pain and redness in her great toe, which is diagnosed as gout. Which medication is most likely to cause the gout attack? Well, this means any drug that actually inhibits the organic anion transporters in the proximal convoluted tubule that excretes uric acid, that leads to hyperuricemia. That's loop diuretics and that's thiazide diuretics. So which one of these is one of those two categories of drugs? Amlodipine is a calcium channel blocker. Acetazolamide is a carbonic anhydrase inhibitor. Chlorthalidone is a thiazide-like diuretic. So it should be chlorthalidone. Which drug is contraindicated in hyperkalemia? Well, that's anything that inhibits kind of the renin-angiotensin-aldosterone pathway. So that'd be ACE inhibitors, aldosterone receptor blockers, and spironolactone. So any of the actual aldosterone antagonists, such as eplerenone. So acetazolamide is a carbonic anhydrase inhibitor, has no effect on the potassium. Chlorothiazide would actually kind of cause hypokalemia. Ethacrynic acid, it's one of the weird loop diuretics, and generally those cause hypokalemia. And then eplerenone is a aldosterone antagonist. So that's actually going to block aldosterone, which will actually cause less potassium excretion. And so that'll cause hyperkalemia. 59-year-old male with the intensive care unit has a metabolic alkalosis. Which therapy will treat this condition? All right. So again, you want something that's actually going to, in a metabolic alkalosis, your bicarb's high, your pH is actually going to be subsequently high as well. So in this situation here, you actually want to excrete bicarb from the body. So a carbonic anhydrase inhibitor or a bicarb excreter to diurese out in the urine would be the best in that situation. So acetazolamide is the only carbonic anhydrase inhibitor in this drug categories here. So I would do acetazolamide. A male patient is placed on a new medication and notes that his breasts have become enlarged and tender to touch. Which medication is most likely is he taking? And so generally, these are going to be drugs that are altering the androgens. And so generally, aldosterone is a steroid hormone. And so whenever you give drugs that are working to block aldosterone at its particular receptor sites, but also has anti-androgen activities as well in other tissue sites, such as the breast tissue, this can lead to these types of effects. So we need one of these drugs to be primarily an aldosterone antagonist, but one that's not a specific aldosterone receptor antagonist. It can actually bind onto other anti-androgen receptors, and that would be spironolactone. So spironolactone is the only one here. Heart failure with a reduced EF diuretics. Okay, so a patient with heart failure with a reduced ejection fraction researched his medications on the internet. Oh boy, found he was taking 2 diuretics, bumetanide and spironolactone. He asked if this is a mistake with his therapy. What is the best response? So you got to go to Dr. Google. So that's never a good idea, but it's a good question, I think. So they are 2 diuretics, but they have 2 different functions. So bumetanide is a loop diuretic, so it's going to help with the symptomatic effects of heart failure with the reduced EF. So in other words, any types of peripheral or pulmonary edema symptoms that they may have, the bumetanide is going to be more successful at. Spironolactone has kind of a couple of functions to go along with that. Whenever they're on bumetanide, they may kind of cause them to lose some potassium, and so we can actually kind of preserve their potassium with spironolactone. It also may give them a little bit of an additional diuretic effect, but nothing intense. But the other thing that we know about spironolactone is that it's actually been shown to reduce mortality, and that's a drug that we want to give patients. If you can reduce mortality, you should do that, and it's also been shown to reduce the mortality by preventing a lot of the cardiac remodeling effects. Remember, it helps to reduce preload, helps to be able to reduce the aldosterone effect on the heart, which causes a lot of the remodeling effect there. So I would say that there is benefit to spironolactone by preventing hypokalemia, giving a little bit of extra diuresis, but on top of that also prevents cardiac remodeling, reduces mortality in patients who have heart failure. So whichever one answers that. Spironolactone is used to prevent hyponatremia? No. It'd actually be used to prevent hypokalemia. Spironolactone is used to reduce heart structure changes and decrease the risk of death. That we know it reduces mortality, so it's got to be B. Bumetanide is used to decrease the potassium loss from spironolactone therapy. That's actually, again, that's the opposite. Spironolactone will be used to decrease that from bumetanide therapy. And this is a duplication error, and only one diuretic should be stopped. No, that's not the case. So I think that, again, the correct answer would be B. All right, last question here. So which diuretic has been shown to improve blood pressure and resistant hypertension, or those already treated with 3 blood pressure medications, including a thiazide or thiazide-like medication? So they're already on a thiazide or thiazide-like medication for their blood pressure, and they're on a couple other medications, apparently. Whatever that may be in addition to this, I'm not sure. But what else could we add on to the thiazide and the 2 other medications that they're being treated with? So indapamide is, again, it's basically a thiazide diuretic, so I wouldn't want to put them on 2 of those, so that would not be the correct answer. Furosemide is not really a true blood pressure medication. If a patient's extremely volume overloaded and hypervolemic, it may help to reduce the blood pressure, but that's not a primary indication for furosemide. Mannitol is not a blood pressure medication as well. It's primarily utilized in patients who have excessively high intracranial pressure. So again, it's not going to be one of those particular situations. Again, in situations where they have high ACP, they may develop a Cushing's triad and maybe given mannitol and it improves their blood pressure, but it's not a blood pressure medication that's designed to reduce blood pressure. Spironolactone actually does have antihypertensive properties. It's not super powerful, but it has some benefit. And one of the good things about this drug is that, it can help with reducing mortality in patients who have underlying heart failure. It may help to be able to block aldosterone, which will help to reduce a lot of the sodium and water intake from the body, which helps to reduce kind of the preload, the excessive blood volume, the venous return, reduce their cardiac output, and reduce their blood pressure. And again, it also gives you some type of resistance to hypokalemia as well. So yeah, I would say that spironolactone out of all of these would be the only one that would actually have a true kind of blood pressure modest reduction, mild reduction. Only reason I can't give him dapamide is I've already given the thiazide-like diuretic. Furosemide is not indicated for blood pressure. Mannitol is not indicated for blood pressure, but spironolactone can help with hypertension, especially if they have hypokalemia, heart failure, or any kind of hepatic edema related issues. So this would be good indications for that. So I would say it would be D. All right, my friends. So that would cover all of these questions on diuretics. I hope it made sense. I hope that you guys liked it. And Ninja Nerds, thank you guys for being so awesome, always sticking in there, continuing to support us. We love you. We thank you. Couldn't do this without you. And as always, until next time.